Well, Redonna, thanks very much. And I want to welcome all of the attendees to this very exciting symposium, where we're going to be hearing science from some of the sort of edge and breakthrough new technologies, new insights into how drugs interact with HIV and ways to use that knowledge, of course, in order to prevent and treat. It displays a group of researchers that have been selected because they were able to get an avant-garde award, which is one of the awards that we give to very promising new investigators that are willing to take risk in the area of science and not follow necessarily the traditional paths that sometimes the regular RO1 mechanisms requires us to do in order to get it the approval. So it has been very successful, and you will see the exciting sightings that you will be hearing today. And before I end, I do want to thank also the leadership of Dr. Redonna Chandler and Dr. Vasundhara Varthakavi, who have basically see the oversight of this program and make it possible. So I hope you enjoy the meeting. Please feel free to ask questions, to give suggestions. This is the way that we can get dialogue in both ways. Otherwise, it just actually will live in isolation. And we don't like that. Science actually very much dies in isolation. It's for everybody. Knowledge is the product of all of us. So having said that, I give it back to Redonna so we can start the symposium.
All right, so let's get right to the science. I'm really excited to see these presentations. Our first presenter is Dr. Ashley Buchanan, and she is from the University of Rhode Island. So take it away.
So good morning, everyone. Thank you very much for this invitation to speak about our Avenir project entitled Causal Inference Methods for HIV Prevention Studies among Networks of People Who Use Drugs. I'd first like to give a few acknowledgements, first to the participants, study staff, and investigators of the 4 primary data collection studies used to motivate this work. I'd also like to thank the following collaborators for their contributions to this work as well. And this work is supported by this Avenir Award. A brief outline of the talk. We'll first introduce our team members, give an overview of our project, share some research highlights, discuss our accomplishments fueled by the Avenir Award, as well as future directions. And I have no conflicts of interest to report. So at URI, our team consists of myself, Dr. Natalia Katanka, who's an associate professor of computer science and statistics, Dr. Hilary Aroke, who's a NIDA Diversity Postdoctoral Fellow, Dr. Tingfeng Li, who is an Avenir Postdoctoral Fellow. This award has also supported multiple graduate students, both in the master's and PhD programs in health outcomes and statistics at URI. We have some key collaborators, including Dr. Samuel Friedman, Elizabeth Halloran, Brandon Marshall, and Will Goodell. And our project officer is Dr. Richard Jenkins. Just an overview of the project. So the project is focused on the population of people who use or inject drugs, who face increased HIV risk through both injection and sexual routes, and unique barriers on the HIV care continuum. At the same time, these individuals are part of communities, risk or social networks where there's an increased risk of HIV transmission. So in this project, recognizing that these individuals are embedded in these risk or social networks, we aim to develop methods and apply them to understand disseminated or spillover effects, which is when one person's exposure affects another person's outcome. So the methodologic problem is that causal inference methods are often challenged by network studies, but at the same time, within these network studies, we want to understand these causal effects. So basically a causal effect is trying to understand if the intervention actually prevents or causes the outcome to happen. So we want to solve these methodologic challenges to address questions at the intersection of HIV and substance use among network-based studies of HIV treatment and prevention among people who use drugs. So this work is motivated through collaboration with different study teams, and we use 2 network randomized studies. These are similar to a cluster randomized study. However, here the cluster is actually an egocentric network. So it's a single individual and their contacts. So we have one site in Philadelphia in HB1037 and another study in Baltimore known as STEP. So in both of these studies, there was a peer education intervention, which showed improvements in reducing HIV risk behaviors. And in our work, we sought to understand the disseminated or spillover effects of these interventions in these populations. We also have network-based studies. So these are either what are known as sociometric or respondent-driven samples. So instead of just the egocentric cluster, in these studies, we have more information about how the individuals are connected to each other or the edges shared between the individuals. So one study is the Transmission Reduction Intervention Project. So far, we've focused on the Athens, Greece site in collaboration with the principal investigator, Sam Friedman. And in this study, they sought to find individuals recently infected with HIV and referred them to treatment as prevention, as well as disseminated community alerts to help individuals realize they may be at an increased risk if there was a recent infection detected in that area of the network. They looked at such outcomes as HIV incidence and risk behavior. The other study we're working with is in New York City among young adults who have non-prescription opioid use. And then here looking at the exposures of such as polysubstance use and the outcomes such as injection drug use or HIV, HCV infection. So a few research highlights. So first, I'll discuss this project in collaboration with Dr. Brandon Marshall and Will Goodell. So in this project, we are using an agent-based modeling approach, which is essentially a mathematical modeling simulation approach to generate this ideal randomized trial. So it's a two-stage randomized trial. And then we can use this model to simulate that trial. And then with the outputs from the model, we can quantify causal effects. So here we're specifically interested in this disseminated effect. So the question we wanted to know is there a disseminated effect of pre-exposure prophylaxis for HIV prevention? And we adapted a model that was among men who have sex with men in Atlanta, Georgia, while also considering whether or not those individuals, if you will, the simulated individuals or agents were engaging in substance use or not. So a little bit more about the two-stage randomized design. So in this design, what happens is we first, in the agent-based model, we first generate the sexual network among the men who have sex with men in Atlanta. And then among that network, we identify what's known as components. So those are individuals that are connected to each other, but not connected to anyone else in the network. So we find those separate components. And then in our agent-based model, we can actually randomize those components to what's known as an allocation strategy. And so that will determine how many people in that component actually are individually randomized to the intervention. Now, the beauty of this simulated agent-based model approach is that it's no longer ethical or feasible to conduct such a randomized trial, given the known efficacy of PrEP. However, in this agent-based model setting with our simulation, we can determine if there's a possible disseminated effect of PrEP. So a little bit more about the different effects that we can identify in this two-stage randomized trial. So we have our intervention components and our control components on the left here. And then within the intervention component, a certain percentage of individuals will be randomized to the pre-exposure prophylaxis, and then the otherwise randomized to no PrEP. So if we look in the intervention component and we compare individuals randomized to PrEP to individuals randomized to no PrEP, we get what's known as the direct effect. So that's the additional benefit to being randomized to PrEP beyond being in an intervention component. We have the disseminated effect or the spillover effect, which I personally find the most interesting in this problem. So we then look at the individuals who were not randomized to PrEP, but in the same component, or at least sharing some path in the component to individuals randomized to PrEP. And we compare those individuals to individuals in the control component. The composite effect gives us the maximal intervention impact. So that compares those randomized to PrEP in the intervention to those randomized to the control. And lastly, if we marginalize over the individual level exposure, we compare just everyone in the intervention component to everyone in the control. So an important connection here is that with the two-stage randomized design, these effects are readily identifiable in the data. So a brief summary of our results from this agent-based model. So on the left, we have the estimated direct risk difference effects. And on the right, we have the disseminated risk difference effects. And this is of the pre-exposure prophylaxis on cumulative incidence of HIV. And then if we move along the x-axis here, this is coverage. So this is the percentage of people or agents, if you will, randomized to PrEP in a component. So interestingly, we see as the coverage increases, the direct effect attenuates somewhat towards the null. And as the coverage increases, we see that the disseminated effect becomes stronger or larger in magnitude, more protective. So interestingly, what's happening here is as we increase the coverage level, the additional benefit to an individual person or agent receiving PrEP, if, for example, 90% of their contacts are randomized to PrEP, would be slightly smaller or weaker. Whereas the disseminated effect, if you're randomized to no PrEP and more people around you are randomized to PrEP, we see that there's a stronger benefit as that coverage increases. So to highlight another project from our group, this one's led by Dr. Ting-Fang Li. So in this work, we developed methods to estimate causal effects of non-randomized interventions in the presence of interference or dissemination in a network-based study. So we're using causal inference approaches to quantify essentially the effects I discussed on the figure a few slides ago among individuals who are embedded in a network, such as people who inject drugs. So for this particular study, we're using the Transmission Reduction Intervention Project in Athens, Greece, as the motivating data. So this visualization on the left, the green dots represent the individuals or the people who inject drugs, and the lines between them represent shared drug use and social connections. So here we focused on, if we look here, I've highlighted what's known as a single component in this TRIP study. And then when we zoom in on the component, just to define what the neighborhood is, we have this is, for example, the individual of interest and in red, and their neighbors are these individuals in orange. So without sharing formulas or too much detail, just to give a flavor of what the method is, we use this neighborhood to define what's known as the interference or dissemination set, so that this person's outcome not only depends on their exposure, but the exposure of the orange nearest neighbors. And then to conduct inference, we identified non-overlapping independent components like the pink component displayed here. So to briefly summarize, we wanted to see if there were disseminated or spillover effects of the community alerts in TRIP. And just to refresh, the community alerts were when the team disseminated information about a recently infected individual near that person in the observed network. So about 11% were exposed to these community alerts from the study staff. And in our analysis, we found there were actually 13 per 100 fewer individuals reporting HIV risk behaviors if 75% of their neighbors received alerts, compared to the scenario where only 25% of the neighbors received alerts. So evidence of a possible spillover or disseminated effect of the community alert information in this particular study. So to summarize some work from Dr. Hilary Aroque, one of his projects is developing causal inference methods to evaluate contamination effects in cluster randomized trials. And he's adapting concepts in mediation to see if there's contamination effects in HP 10037. So recall HP 10037 was the peer education intervention among people who inject drugs in Philadelphia. In the study, this intervention was effective for reducing injection risk behaviors. In a subsequent study, we did find and quantify disseminated effects of the intervention, but still contamination remains a concern. So what was the intervention actually reaching individuals in the control, or were those clusters actually interacting with each other? And through this work, we have landed upon contamination in this setting is actually only meaningful if we can demonstrate that the intervention is actually mediated through the intervention knowledge. So we were going to quantify the contamination to see if the control clusters actually had knowledge of the intervention. And for that to be meaningful, we first have to see if the intervention is in fact mediated by that intervention knowledge. So first we're looking at mediation, which is to decompose the total effect of the intervention on self-reported injection risk behavior into that mediated through the recall of intervention knowledge, as well as the remaining direct causal effect through all other mechanisms. So we use the information at 6 months to assess the patient recall of the intervention knowledge, and at 12 months, assess their self-report of injection risk behavior. In our preliminary findings, only about 5% of the total intervention effect was mediated by intervention knowledge, which tells us there could be other pathways, and we suggest that this may be pathways such as behavior modeling. And just to briefly mention some work in progress, Tinyu Sun is a PhD candidate in our group, contributing to this work to use a multi-state model to investigate the opioid use disorder care cascade in Rhode Island. So there's been ongoing work to try to define the opioid use disorder care cascade, as well as several papers looking at cross-sectional assessments for risk factors. So our group recognized that the cross-sectional assessments are helpful, but there's actually an underlying longitudinal process by which individuals are moving through this cascade. So we're going to develop and apply a multi-state model to look at factors associated with both transition and barriers along the OUD care cascade. And this is using the Rhode Island All Payers Claims Database. So here's the conceptualization of our model. Essentially, we're focusing on the engagement and retained in care piece, as that is more reliable information from the data source that we have. So we're looking at first engagement, short-term retention, and then long-term retention, and then also how long patients are disengaged. So just to highlight some accomplishments of the team so far, I'd like to mention some opportunities provided by the Avenir that without this support, I don't think our team could have made progress so quickly. So our team is uniquely positioned, having a person specializing in network science as well as causal inference with a focus on substance use research. So I think this uniquely positions our team to be working at both the intersection of HIV and substance use, as well as network science and causal inference. The Avenir has led to invited talks for multiple team members, which all resulted in new collaborations and including access to important data sets, which is incredibly important for the biostatistics methods work. We like to use the studies and the data to actually motivate the methods development, and then the methods development can then in turn provide better guidance and information for design of future studies. We are also delighted as a team to help support open access to some of the data sources that were given to the URI Avenir team, which I personally find to be a very important scientific endeavor. The Avenir also has created incredible research experience for the graduate students, working with principal investigators and incredibly rich data sources. It has also provided a mentoring platform for postdocs with opportunities for training in grant writing and speaking engagements. Our team also gets to meet regularly and collaborate with key researchers in the field, which has greatly improved our work. And it has also opened opportunities for the development of didactic lectures and materials for the research coming out of this project. So just briefly, future directions. So as I mentioned, there's a synergy in our work between the methodologic developments and the substantive area of substance use and HIV. And I see the two working with each other to move along. So methodologically, we're interested in areas such as missing data and longitudinal in the context of dissemination and interference. So looking at things like a time-varying interference set. We're interested in looking at some additional modeling approaches or Markov random fields to work on further methods development in this network-based study. And then, of course, this is related to substantive questions. Our team's interested in addressing issues related to the syndemic of COVID-19, HIV, HCV, and the opioid crisis, as well as addressing racism and health disparities. Specifically, they're developing a project to do some COVID-19 modeling that incorporates network structures to help address health disparities. And also, of course, continuing to work on the OUD care cascades, synergizing definitions, developing new methods so we can better improve the inference that we're getting from these routinely collected health data sources. Oops. So in conclusion, in many cases, and so far what we've seen in this project is dissemination and network structures seem to be substantial and possibly meaningful. And I see this as good news so that many NIDA funded studies and other interventions could possibly have higher benefits for public health than standard techniques reveal. And I think, our team is very much interested and excited about this and different questions to try to understand what these disseminated effects are and how we can better use them to help improve the uptake of interventions in this population. So a few recent publications from the team and some publications in preparation. So thank you very much for listening. That's all I have for today, and I'd be happy to answer some questions.
So Kavi is going to proctor our questions. If people have questions, you can type them in the chat or in the Q&A, and then she'll read the questions for Ashley to answer. We've got about 10 minutes or so to have a robust discussion and some Q&A. So Kavi, you ready?
See any questions here? I'm looking up the chat. Also, we can unmute if anybody wants to ask questions.
Kavi, I don't think that the participants have the ability to unmute. I think we're going to need to read questions that they type in.
Yeah. Don't see any coming in. Okay, there is.
If you'd like to also ask a question verbally, you can raise your hand and then we can know to call on you and open your line up momentarily.
So there is a question in the chat from Karen Ingersoll. She says it's very encouraging data. I think the question is, how much are we underestimating the effects?
I think that's an excellent question. I think, this is somewhat of a vague answer, but I think it somewhat depends on us going back and looking at studies. We can look at studies that have already been conducted and see, what is the magnitude of that disseminated or spillover effect, and how does that compare to the overall? So I think it may depend on the study context, but I think, moving forward, our team is also collaborating. I think this is very important, particularly for study design. We're working on sample size and power calculations to help inform the design of future studies, so that if investigators are interested in quantifying this, we can make sure you have enough power in your study to do that. So we could be, maybe in the future, if there's a possible, I think the important thing though is that there could be a possible disseminated or spillover mechanism, and making sure we know what that is first. And then if we suspect that could be there, why not power and try to estimate this?
Thanks, Ashley. I don't see any more questions here. Oh, there's one more. This is from Dinesh Kumar. Does the method or tool to measure contamination have effect over assessment?
I'm not sure I follow that question. So maybe I can make it a little bit clearer. So today I focused primarily on discussing what the mediation was that we found. So the contamination it's like we're basically estimating an effect that, if that's protective or harmful, it will tell us if there's possible contamination happening in the study. And then I guess there's sort of two pieces with that. One piece is, you first want to know, is there contamination or not? And then the second piece would be, do you want to adjust your estimated effect to account for that contamination or not? So those are sort of two separate questions I see in that work. So maybe that was what the attendee was asking.
All right. He's trying to explain. Okay, effect over results, he says. Aaron, can we unmute Dinesh? If he can speak, we have time.
Thank you for giving me the opportunity to ask question. What my question is, I'm sorry, I was not clear. Does the way we are going to assess the measure the contamination, like asking question from the person, or whether it is a interview based or record based or self-reported, whether such kind of an assessment has impact over the results, that this much has contributed to indirect effect and this much has contributed to direct effect or direct contamination. So that's my questions. Thank you very much.
Yeah, I think that's definitely an important consideration. So for this initial work we're doing in the contamination effects, we're basically using a study that's already been conducted, and this was the information they collected to see if there was contamination. It seems like it could be very wide open to figure out, how can we measure and quantify contamination? What's a valid way to get at that for these different interventions? But in HB 10037, the recall of specific terms from the intervention was how they measured contamination. And of course, that would definitely impact, how we're measuring that, and the quality of that would impact our ability to assess contamination.
Thank you. Thanks a lot.
Okay, so there is another question from the audience. It says it's a general question regarding future goals for understanding impact of racial, ethnic, gender bias, social determinants of health, including healthcare access. Is there data that you wish the community would be collecting?
Oh, so like the research community.
Yeah. I don't know. Maybe the community in general that is part of the study.
I mean, I guess one thing I've noticed in my work with routinely collected health data, and some of this is administrative claims data or even the all payers data, I would encourage us, as we move towards continuing to work with health disparities and quantifying those, that if we have Department of Health data, for example, if we're able to collect race and ethnic information and other factors like that to help better understand the disparities in these data sets. So I know the Rhode Island Medicaid does collect race information, but then working with the Rhode Island all payers data, it actually doesn't have the race information. So we're actually not able to even dig further into possible health disparities about the OUD cascade in that data set. But that's routinely collected data. And then I'm trying to think it sort of depends on, like, moving forward for primary data collection, what would be of interest in the study and additional information to collect to understand disseminated effects.
Thank you. Well, looks like that's all we have. So thank you very much, Ashley. That was a fantastic presentation.
Thank you, everyone.
Okay, so we will move to our second presentation from Dr. Daniel Lingwood, and he is from MIT Harvard.
Good morning, everyone. I'd like to start by thanking the organizers for the chance to present some of our work that was is and has been supported by this Avenir Award, and how we think it's really been transformative for us in terms of actually introducing us to the HIV field as somewhat of an outsider. So the title of my talk today is called Epitope Shifting to an HIV Vaccine Target Through the Acquisition of Low Affinity, Applying the Activity of an Innate-like Immune Receptor. And I hope it'll become very clear what all of that means as we play out. So just by a brief background, just to start off with, of course, the development of ART has become a transformative, life-saving intervention. However, I think as this audience is well aware, much more so than I am, that the number of individuals, including in this country, who are diagnosed with HIV, very many of them are still left out of the system in terms of being left off ART, and there are socioeconomic, behavioral barriers that complicate this adherence behavior, and I think it's well appreciated that this is particularly problematic in substance users, and so this sort of strengthens the notion that an HIV vaccine would particularly be amenable to improving the life of these individuals. And so one of the fundamental questions that I guess one can consider is what are the challenges to generating such an HIV vaccine? Why is it, for example, that we can't roll out a vaccine as fast as, for example, the coronavirus SARS-CoV-2 has been done? And the simple answer to that is the diversity challenge. So HIV presents the immune system with something that is sort of never been seen before in terms of a diversity challenge. So if we look at the genetic diversity on the left here, from a total global influenza epidemic, we can see that it's actually comparable to the HIV diversity that occurs within a single person, and that's just orders of magnitude higher when we look at sequence diversity in the HIV space across the world. So it represents an unprecedented challenge to the immune system. How do we develop a vaccine, a set of molecular instructions which teach the immune system to sort of keep up and constantly be able to neutralize this incredibly diverse virus? If we look at sort of the problem at the sort of immune response or antibody response level, following HIV infection, most of us would develop what we will call type-specific antibody responses. So the blue antibodies neutralize the blue virus, the green virus, the purple virus. And if we sort of blow up that type-specific neutralization, those antibodies are sort of intrinsically directed towards those hypervariable parts of the virus, which are, as I say, are constantly evolving and contribute to that global diversity problem. However, there are rare cases when certain individuals produce what I've titled here in the genetically encoded broadly neutralizing antibodies, which under these conditions are able to reproducibly target some of these conserved regions and neutralize much of that global diversity. What do I mean by genetically encoded? Well, for each individual, of course, we're made up of genes on our chromosomes, including part of this package. We come equipped with these so-called antibody VH and VL genes, antibody V genes, which go into making up the diversity of our antibody repertoire and our ability to respond to different incoming antigens. And so some of our work in the past has looked at how certain V genes, some of these antibody genes, naturally produce certain kinds of antibodies which have this sort of natural affinity for functionally conserved sites of vulnerability, which viruses like influenza are functionally constrained to preserve. And this endows humans with a kind of reproducible gene-encoded antibody response. And so some of our work both in the past and ongoing, has been focused on trying to harness this principle as a means to develop universal flu vaccines. So what the Avenir has done is allowed us to explore this similar concept in the HIV space. Now there is this analogous antibody VH gene. I'm going to refer to it as the HIV gene. I'll specify a little bit more later, which is also in these rare individuals reproducibly used to create broadly neutralizing antibodies against HIV, in particular that target a functionally conserved feature on the virus, which is called the CD4 binding site. So CD4 is the receptor HIV uses to engage cells. This HIV gene endows for antibodies, which engages this target in a very precise way that enables neutralization of over 80% of that global HIV diversity that we were talking about. So the notion is potentially there is this analogous gene endowed antibody response that may be amenable to sort of vaccine activation and amplification, akin to some of the work that we've done with influenza. And that was really what the Avenir allowed us to now explore and define in the HIV space. And so basically, the overall topic that we've been working on with this under this award is the question: how does this so-called HIV antibody VH gene actually work as an immune receptor? And there are two sort of stories which I would like to focus on during this talk, which I think have sort of been the most exciting discoveries of our work in this space. And number one is so why would humans be equipped with such an HIV gene that sort of reproducibly endows for this broadly neutralizing antibody response and sort of potential template for HIV vaccines to sort of conquer that diversity? And the second question is, how do we actually activate that gene encoded and potentially amplification principle in the vaccine space? So in order to answer these questions, starting with the first, why might humans be equipped with this VH gene? What we're dealing with, the question really is, what is it about that gene that endows, in this case, antigen receptors, in this case, what are called germline B cell receptors, which are the membrane anchored forms of antibody precursors, which are present on the surface of your germline B cells that are in your spleen and your lymph nodes. These receptors are charged with initially recognizing incoming antigens. So whether it be on an infecting virus or in a vaccine, defining the geography of that we say epitope targeting, and then that's gonna as I say, define how and where a given antibody response is going to target. And so sort of the question that we're asking is what is this HIV gene playing with respect to that? So if we blow up this antigen receptor a little bit closer and look at the molecular goings on here, we can think of each B cell receptor as a series of fingers which grab the antigen. So these fingers are made up of V gene encoded loops, and also essentially positioned highly variable random antigen binding loops. And it's these V gene encoded loops which the antibody V genes, including this HIV gene, are sort of playing into. So we want to understand what this red gene is doing with respect to these gray loops, which is somehow predisposing for recognition of this vaccine target. Now, so in order to experimentally interrogate this, we had to actually build a system where we could do that. And I just say that I'm going to finally name this HIV gene. It's called IGH-VH12. So I'll be referring it to that throughout. But in terms of the experimental system that we built, we had a collaboration with our industry partners at BMS to sort of build a system where we could experimentally ask, what is the contribution of a given antibody VH gene to antigen recognition and the immune response in the context of an experimental transgenic mouse system? So we built this transgenic mouse system, which basically recapitulates all of the human antibody loops, in particular this hypervariable CDRH3, but at the same time systematically restricts antibody responses to a single user-defined VH sequence in the animal. So basically, we can perform an experiment. What is an experiment? You fix one variable, vary something else, and assign causality to the variable you fixed. So essentially what we're doing is we're preserving the hypervariable
Sorry, there is some technical glitch here, so we're waiting for Dr. Lingwood to get back online. Please be patient with us.
Well, while we wait, I want to call everyone's attention to the fact that if you have not applied for the Avenir Award and you're interested, Kavi has posted information in the chat that everyone should be able to see, including the link to the funding opportunity announcement. And please feel free to reach out to us at any point in time where you have ideas.
Hi, apologies for that. I'm not sure what happened. Let me just quickly start this again.
Yeah.
It happens to everyone.
Yeah,
And always at the most opportune moment.
Yeah, let me just go back to where I was. Is that, can people see that?
Yeah, we are good.
Yeah.
Okay, thank you. Again, apologies, my computer froze. So basically, just to quickly summarize, we built an experimental system which basically allows us to fully humanize and mimic that hypervariable middle finger loop, but systematically preserve and control the V gene input, that gray input to the surrounding antigen binding site, and then ask experimentally, what is it doing with respect to antigen recognition, vaccine targeting? What is this so-called HIV V gene actually doing? And so just a bit of characterization of these animals. We can look at that hypervariable middle finger loop and see, in terms of what we call it the CDRH3, we can see that it's matches the human repertoire in terms of length distribution and amino acids that go into that loop structure. But again, we're constraining the system to use individual single V genes at a time across the entire animal. So coming to this question, why might humans be equipped with this HIV V gene? And so, you know, it's unlikely that we sort of evolved this specificity recently. HIV is a fairly new virus. We sort of said, well, what is it doing with respect to antigen recognition, particularly at the level of primordial antigens, things that would have been present in our past. And what we thought was, first, let's look at some bacterial antigens. These are sort of ancient immune stimuli, which we have come equipped with a very sensitive, rapid response system in our blood that's tuned to recognize these targets. So if you put either bacterial sugars or lipids into your blood, they're very potent septic signals. So you get a very rapid antibody response in your blood to clear those antigens, target those bugs. Of course, bacteria presence in the blood is very dangerous. It's very easily measured by what we refer to as a extrafollicular IgM response. And so we said, okay, these are some primordial antigens. Here's a response system which we can use to gauge microbial input to this. What is this HIV gene doing in this context? And so what we do is we take our transgenic animals, which is using our HIV gene here, VH12, a control human gene, a different human VH gene, VH169, and we can put in a couple of control antigens, sugar polymers, TMP ficol, ovoalbumin, and we can see that we get this rapid extrafollicular response coming from the spleen in all of our animal subsets. I think that the interesting discovery that we made in this context was that if we put in bacterial lipopolysaccharide, which is the major surface antigen of gram-negative bacteria, so we put that in covering diverse strains of bacteria over thousands of years of evolution, over 2 orders of gram-negative bacteria, what we find is that it's only the animals that are using the HIV gene that are able to accommodate antibody responses to this bacterial antigen. If you're using a different V gene, despite the fact that you've got all the diversification machinery working for you, you're unable to recognize and respond to that. And so, sort of to make a long story short, we did some work to actually show that this HIV gene was tuning the germline B cell repertoire to respond to a conserved part of this antigen, the so-called lipid A core of these bugs, which doesn't change. And so what you've got is this HIV gene, which is naturally tuning the immune system to recognize a conserved microbial target. So kind of like an innate-like immune receptor system, but for the adaptive immune response. And so we thought that was quite an interesting, at least functional perspective to think about why it is that we have this VH12 in our human repertoire. Why is it useful? It endows the immune system with a broad spectrum antibody response targeting this conserved hydrocarbon core of an otherwise highly divergent form of bacterial LPS. So again, this innate-like immune recognition. Okay, so we've got sort of a functional idea with our system as to why, as to some speciality of this VH12 HIV gene. How do we actually now make it work for us in the HIV vaccine context? We know that it somehow predisposes for this neutralizing activity. How can we now harness that? And so we go back to this transgenic animal system that we've built, where we can focus antibody responses, constrain antibody responses to use this gene, but still let them have the appropriate diversity play to explore the antigenic space across a potential HIV vaccine. So what we decided to do was to sort of apply a sort of non-conventional vaccine strategy to think about ways in which we can immune focus the attention of the antibody response on this conserved site of vulnerability, the CD4 binding site on HIV, all in this VH12 context. So if we think about, this is here a picture of Edward Jenner, 200 plus years ago. We have the sort of conventional form of vaccination where you have a prime, we call that injection 1, a boost. So those of you who are getting your COVID vaccines, you know, you get a prime and then 3 weeks later you're scheduled for a boost. This is a very classical vaccine paradigm. So however, in the context of HIV, which of course is immune distractive, the prime will likely give us a lot of antibody responses that are targeting the hypervariable blue part, right? And then when we boost that, we simply boost the immune memory against the blue. And then we sort of call that a homologous immunization. So that's unlikely to work for us in order to refocus the attention of the immune response on this red conserved part. However, if we take a different approach, we start off with the same with the blue immunogen, we'll get a response against the blue, maybe a little bit against the red. But what if we take advantage of the sort of existing HIV diversity that we know and now hit it with a different target, a different strain of HIV, which is functionally conserved to still hold the red, but changes the green? Maybe we can refocus part of the antibody response. We're basically showing the immune system 2 different entities, but the only thing that's conserved is the red. So maybe this heterologous immunization concept can help us refocus to the red. And so this is what we ended up doing in our vaccine model, where basically we came up with a number of different strains of HIV, envelope strains of that display that conserved target and otherwise varying background, and sequentially immunized our transgenic animals. So if we sequentially immunize with a homologous, where it's just we're doing blue, 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 we can measure the antibody response. We're looking at IgG titers against either the wild type form of this blue immunogen or the D368R form, which basically blocks access to this CD4 binding site. So there's no delta between the curves. However, if we now apply a heterologous immunization scheme where we're constantly changing the varying the antigen, we can now refocus part of the immune response on this otherwise immunologically invisible target. So we were quite excited about that discovery. And so we said, okay, what molecularly, what is going on in terms of how this HIV VH1/2 immune receptor is working? We noticed something very interesting. If we sort of reperform these assays in the presence and absence of urea, so urea is a chaotropic agent which basically destabilizes interaction. What we looked at is resistance to urea in the homologous scenario versus heterologous scenario. What we found is that when we're refocusing to the CD4 binding site, what we find is an increased resistance to, or a decreased resistance to urea, suggesting that when we're refocusing the attention of the immune response to this target, there's an affinity drop that's happening. And this was sort of interesting because conventionally, when you sequentially immunize, there's not supposed to be any affinity drop. You're supposed to gain in affinity. So just a quick review on that. So step one, when you immunize with an antigen, you come in. Germline repertoire engages the target as we discussed, defines the target epitope. There's a process called somatic hypermutation that takes over that increases affinity to that target. But what we're seeing is this affinity drop when we refocus the attention of the immune response to this putative HIV vaccine target in this VH1/2 context. And so we wanted to explore the molecular basis for what might be going on. We have a way to, we developed a way to actually isolate the antigen-specific B cells that are underscoring the vaccine response. So we can basically define using a set of fluorescent B cell probes the on-target antigen-specific B cell memory that's targeting this CD4 binding site versus the off-target responses, which are seeing elsewhere on the target epitope, and then we can reconstitute them in terms of BCR antigen interactions, which we developed in our lab. And so what you're looking at is a BCR antigen interaction readout, where basically what we're doing is reconstituting the BCRs that we vaccine expanded and asking which of our vaccine immunogens across the top did they see and how did they see it. So for example, as a positive control, we look at VRC01 BCR. This is a classical broadly neutralizing HIV, human HIV broadly neutralizing antibodies, and not neutralized 80% of circulating strains. What you can see is that when we look at how these vaccine antigens bind each different component, we see that it's binding to the wild type, but not the D368R of YU2. So we know that it's a focus to the CD4 binding site. Same with the turquoise immunogen, same with the red immunogen, same with the blue immunogen. If we look at S1, which is in a lineage we expanded in our vaccine animal system, we can see, if you just look at the bottom, the mature response, the mature BCRs that we've expanded, we can see that we're binding to, we're recapitulating the same CD4 binding site specificity. We're binding to the wild type of the yellow, the wild type of the turquoise, the wild type of the red, and the wild type of the blue. So we've expanded what looks like to be a VRC01 like antigen specificity. The interesting thing is when we look at the germline precursor, so what gave rise to this S1 lineage, so what started it all off, we find that there's only antigen specificity for the blue antigens. So it was the blue antigen here that triggered the response when we look at the germline response. But unlike the mature, which is binding only wild type and not the mutant form, we find that it binds both forms. So it's saying that this process was actually triggered by an immune response that targeted not the CD4 binding site elsewhere, and that it was only following application of affinity maturation that this blue went away and we're targeting the right specificity. And so we can actually map out that process by looking at the pattern of somatic hypermutation that went on during this immune response. And basically, to make a long story short, we reconstitute some of those specificities, and we can see that we start out from germline. We're binding both immunogens, wild type versus D3CD8-4. So we're starting off target, and then only after application of subsequent affinity maturations do we switch to see the CD4 binding site. And the interesting thing that we see with regard to this switch is that at the mature stage, when we're switching this, if we actually look at the corresponding biochemical affinity between antibody, which is part of the B cell receptor, that is just the single FAB binding arm, and the target, what we find is that there is this affinity drop scenario. So basically, we measure this using bilayer interferometry, where we look at the KD. So a high KD means low affinity, a low KD means high affinity. So we see that the KD starts at around 1.5 micromolar, comes along. It's only suddenly when we go from off target to on target that we get this huge affinity drop. And again, remember that we have this polyclonal response pattern where we're again seeing this affinity drop in the refocused antibodies across the board. And I should say it's not just this lineage that we're seeing this in. This is just a representative example. We're seeing this affinity drop come time and time again. And so what we think we found using this targeting system is a sort of a new way of thinking about how antibody somatic hypermutation participates in the epitope discovery process. So the conventional view, again, is that the germline BCR defines the target, and then somatic hypermutation increases the affinity to that target. What we found in terms of understanding how this special VH gene works in terms of being able to see this CD4 binding site is that it can start off with an irrelevant target that's initially engaged, and then it's only in the context of affinity maturation that you don't necessarily, or somatic hypermutation, that you don't gain affinity as is sort of convention. You don't have to gain affinity as conventionally thought, but somatic hypermutation can actually act as a further diversifying agent, which sort of laterally explores the antigen and can acquire specificity for this target, and this system sort of enables this discovery of what we would call this sort of affinity constraining target. And once that is then established, our hypothesis and sort of next steps is to sort of define how now we can push that engagement along towards broadly neutralizing activity. So I think very generally, just to summarize, I'm a sort of influenza B cell biologist who was able, thanks to the Avenir Award, to now enter the HIV space and define what I think are two fundamentally new sort of contributions to our understanding of immune recognition. We know that this VH gene is reproducibly used in a subset of humans that give these broadly neutralizing antibody responses. We have found that this HIV gene is sort of a natural and innate-like immune receptor for microbial antigens, suggesting maybe that microbial antigens actually play a role in priming some of these responses. And with respect to question 2, in terms of understanding how we activate this receptor biology, we've defined a sort of non-conventional mode of antigen complementarity, a new role for antibody somatic hypermutation, which enables these antibody products to sort of engage this affinity constraining target and acquire that specificity. So we're very interested now in thinking about ways in which we can potentially harness this further in the vaccine context. So I'd like to end it there by again thanking the Avenir Award, which supported this continues to support this HIV program in my lab. It's supported in particular two postdocs, Ashraf and Lawrence, grad student Aventis, who has all coordinated or worked heavily on this on this area, our industry collaborators, and thank you to the Reagan Institute, and thank you for your attention.
Thank you, Dr. Lingwood. We have some questions coming in the chat now. So here is one. What are the implications of your findings for germline target vaccine strategies?
Yeah, so I think what we've sort of found in terms of, so the germline targeting notion is, is to sort of engineer a high affinity immunogen so that you can sort of actively select those sort of correct germline precursors of interest and then expand them. I think what we've sort of found is we haven't used a germline targeting principle in this case. I think what we found is we're more along our discovery is more along the lines of defining a natural way in which the immune system is able to perceive these low affinity BCRs, that there's actually sort of inbuilt flexibility in terms of a non-homogenizing affinity selection principle at play that enables low affinity BCRs to subsist for long periods of time. And I think this is more informing of the natural way in which this target is perceived by the immune system.
And there's a follow-up question here. How problematic do you think existing B cell repertoire will be? Does that constrain how much you can educate the system?
Does that mean, can I just ask, does in terms of pre-existing immunity to other things? Is that the question?
Um,
Can I use the fact that I'm a panelist to clarify rather than type?
yeah, if you
Sure.
can unmute.
Daniel, maybe I'm speaking right to your wheelhouse, right? Because the the thought that pops into my mind is like original antigenic sin and like its issues in
Sure.
flu vaccine. And now, okay, most people haven't seen HIV antigen, but I wonder if that kind of effect might, be
Sure.
something you have to overcome.
I think you're absolutely right in terms of so I mean, number one is as you point out that most individuals obviously haven't seen HIV, unlike influenza. So the notion of pre-existing potentially distractive immunity in the context of sin would be less of a problem, is my sort of 60,000 foot answer. But in terms of individuals that, for example, have seen HIV or have been vaccinated with some of these concepts, I think that's probably where the notion of heterologous immunization is potentially gonna help play into this, where you can sort of display variant antigen and boost on conserved sites. That would be sort of my sort of generic answer to that problem. But I think, like the influenza situation, it's complicated because it's going to be slightly different for each individual depending on the strain of HIV that they've seen, and so it does complicate a sort of universal solution to this.
Okay, so we have, I guess, a question from NIDA, Dr. Chandler, and myself, like, have you looked at this model in the presence of opioids? In that, how do they?
Right. So I mean, we haven't, we've just basically trying to understand some of the fundamental principles of targeting, but that's certainly a direction that we'd like to go. I mean, I mean, that's sort of one of the I would say an important next step. I think really what we've concerned ourselves with is sort of really making sure at this point, because we've found a result that is sort of contrary to what we would predict based on how we understand antibody affinity maturation to work, we've really tried to be comprehensive in sort of assuring ourselves that this mechanism really is the way that we think it is. And then sort of saying, okay, how can we come in and perturb? But I think, you know, in terms of asking, you know, what are the consequences of exogenous parameters, because I think we, as I say, we have this now unexpected finding that says to see the way that, at least one way that these natural immune receptors work, it's backwards to what B cell immunology might say. And you know, we really wanted to be sure that that's in fact the case before we can sort of now add parameters and say, okay, well, if we now put these animals on opioids, how does that refocusing now change? That's certainly, but that's certainly something that's on our radar.
Absolutely, thank you. So we have a question also from the audience. This is from Siddhapa Reddy. So is this immune focus vaccine strategy will be beneficial to HIV infected drug abusers? How and why as compared to non-drug using individuals?
Yeah, I think I think that sort of speaks to sort of two of the previous questions. Number one is the potential for antigenic sin, i.e., if you're already infected with HIV, there is you know, that you already have immunological memory against HIV, and so one needs to understand what heterologous immunization principle you can apply to that scenario. I mean, I think now that we have kind of proof of concept that under certain experimental conditions, we actually can get the immune system to see this target. I mean, that was really the first question, is can we actually do this at all? Because if you just simply immunize with HIV vaccine antigens, you don't get antibody responses against the target. So we had to sort of establish a baseline to say, or really work hard to define a way in which we can actually achieve, number one, bulk polyclonal antibody response refocusing to the target. Now that we can do that, I think it's amenable now to explore the more complicated parameters where okay, you're on if for a substance abuser, what is the impact on the antibody response titer, the ability to do this. If you're pre-infected with HIV, that will sort of change the B cell memory landscape upon which you apply the vaccine regimen. We've actually done a fair bit of work sort of asking, you know, what if we change the order of how we do the heterologous immunization, we actually see different effects in terms of the ability to refocus the antibody response on the target. So that sort of may play into the kinds of or the ability to sort of apply this in a context of pre-existing immunity to HIV. Yeah.
That's great. We have a question from Dr. Volkow. She's asking if there are known polymorphisms for the HIV gene, and if so, how do they affect responses to HIV?
Sure. So the answer is yes. There are a number of alleles of in this case VH12. The allelic variant that is sort of the so-called HIV gene in this particular case is called VH12. And so that's generally ubiquitous across the global population. So I think I don't know of any reports suggesting that that humans are not equipped with this particular allele. I think one of the interesting features of these so-called VRCO1 class responses that flow through this antibody VH gene, they're all using this very specific allelic variant, VH12. And so that is actually the allelic variant that we used to generate our animal model.
All right. Let's see if there are any more questions coming in here.
I think we have time for maybe one more quick question, Kavi, and then we need to move on.
Okay. So, yeah, I think we'll just move on to the next talk. Thank you, Dr. Lingwood. That was a fantastic talk.
Thank you. Sorry about the freeze.
No problem. That's the world we live in now.
And so next we have Dr. Ryan Westergaard, who will be giving us a presentation, and he is from the University of Wisconsin. So Dr. Westergaard.
Thank you, Dr. Chandler, and for the organizers for putting this together. It's a tremendous opportunity to be able to share this work with you. I'd like to start by acknowledging a really hardworking team that has done this, that is shared in this project with me at the University of Wisconsin, Johns Hopkins University, and our community-based collaborators, an organization called Livent Health, which provides HIV care and prevention services. One of the things that the Avenir program has been really helpful for is building such an interesting interdisciplinary team. And it's been fun to work with people who are in industrial engineering and mass communications, and among other fields I don't get to work with. And in particular, from my perspective as a physician who sees HIV patients, the Avenir Award Program has really been a privilege in that it provided an opportunity to focus a major part of my research on a specific and enduring challenge that has caused a lot of frustration for those of us who see patients in HIV care. And it's the fact of disengagement in HIV care and where and when that happens. And to describe what this challenge is, I made this little cartoon. The main determinants of poor engagement in HIV care are social and behavioral factors. And in every HIV care setting that I've worked, and most are funded by the Ryan White Care Program, we have a robust set of resources that address social determinants of health and behavioral determinants, alcohol and drug use, counseling, housing assistance, medical case management. These things are well developed and meet a lot of the needs, but they happen over here on the right side of this graph. They're embedded in clinical settings where people essentially have to show up and be engaged in order to benefit from them. The threats to care engagement happen over on the left side here in the community, and we don't have good information about when and where these threats are occurring. So that is the challenge that our work tries to address. The implications of this challenge for our goals of ending the HIV epidemic or meeting goals like the World Health Organization 90-90-90 goals really is highlighted by the data among people living with HIV who have substance use disorder. And this slide shows data which are a little old, but still highly relevant to the problem I'm discussing today. The continuum of care among people who inject drugs shows that cross-sectionally, they seem to do pretty well. These are data from the ALIVE cohort in Baltimore, which shows that of people who are HIV diagnosed, greater than 90% are linked to care, most initiate antiretroviral therapy promptly, and most of those have an undetectable viral load. But when we look at the issue of sustained viral suppression, which is really what's necessary to prevent onward transmission and the foundation of our treatment as prevention paradigm, it's actually greater than 90% who will fail if you follow them long enough. So only 8% after they become virally suppressed remain virally suppressed through the follow-up in this study. And that is not unique to this population. So there are ongoing threats to medication adherence that are complex. Some good news, our ability to predict which patients in our clinical settings are at risk for poor outcomes comes from studies like this. This is a predictive model using data from electronic health records at the two Harvard affiliated hospitals HIV care programs that showed using data from EMRs with some basic characteristics that are derived at baseline, meaning at entry to care, we can risk stratify people. And so people who are at high risk, people that had I think 4 or more of these 7 counts, 7 variables, within 2 years, 50% of them had failed. So it's not a mystery who is at risk. The challenge, the mystery is where and when over the course of their illness course is the threat going to be greatest, because our ability to intervene and to support people when and where they need it depends on us understanding that. So that's really one of the research questions that drives the work that's been supported by this award. What are the time varying determinants of lapses in HIV care, and then subsequently, what can we do about them? This is some other background work before this study, also from ALIVE, which addressed this question. The benefit of using the ALIVE study is that it's a community-based cohort of people who inject drugs, who have HIV and mostly been linked to care and have good access to treatment. What we did in this study was look at paired visits, so visits where they were assessed in people who had viral suppression or undetectable viral load. And we looked retrospectively in the data to subsequent visits when they were not suppressed, meaning they had treatment failure. And we looked at what social and behavioral factors were captured through the battery of questionnaires that could give us insight into what are the main drivers of that. And these were the top 5. The number one by a substantial margin was incarceration, particularly short stays in jails, which had a really disruptive effect on medication adherence and was the strongest predictor of virologic failures. The other were stimulant use, unemployment, homelessness, or any injection drug use. So these things, by contrast to what we saw in the Robbins paper from the electronic health records data, are typically not contained in patients' charts. And we need to go elsewhere. We need to have a window into the sort of lived, the daily lives of patients if we're going to understand when and how these things are acting. So this is kind of where I'm leading. If our goal to meet our ending the epidemic goals require novel strategies to prevent and mitigate lapses in HIV care, what do those strategies need to address? And I put them here in two categories. The first is that, as I said, threats to HIV care engagement occur out of view from the providers, and providers I mean broadly, who are in HIV care settings, who are sort of dedicated to help them. I could call this information gaps. The second piece is that the addiction and the disruptive life events that are associated with it, including unstable housing and decompensated mental health conditions and so forth, often compete with HIV care-seeking behaviors for priority. So when people are struggling with these other things, they feel less motivated to engage in HIV care because they have other things that they're struggling with, and we'll consider this a motivation gap. So the tool that we're studying in this research to try to address these two determinants is the CHESS mobile health application. CHESS is the name of both the mobile health app and also the center, which is at the University of Wisconsin. They have a P30 NIDA Center of Excellence grant, and David Gustafson, who is the founder and still director of CHESS, has been a collaborator and a wonderful mentor for me through this work. And they have been doing this work since before HIV, frankly, and way before smartphones, using home computers, information communication tools to try to get people feel more engaged in the care that they received and have greater autonomy into self-management of these conditions. And more recently, there's developed a commercialized version that has helped in the scalability of CHESS-related tools. And there's now a private company that has served larger systems of addiction treatment, including the state of West Virginia has a contract for this. So it's been used, and this is an evidence-based strategy for addiction relapse. And the innovative piece of our Avenir Award funded program project is to try to understand what is highly impactful about this and translate this to the HIV care setting. The framework, the theoretical framework behind CHESS is self-determination theory, which holds that when it comes to health-related behaviors, people are more likely to engage with them if they are intrinsically motivated to engage in the behavior, much more so than if they have an extrinsic motivation or they're not motivated at all. And intrinsic motivation is supported by 3 things: perceived competence, perceived autonomy, and the sense of social relatedness or connectedness to other. So the CHESS app, and it's taken different names here. ACHESS, it stands for the Addiction CHESS platform, incorporates different services to try to support these 3 things among users. we, to distinguish it from the HS or addiction chest, we called our study, or nicknamed our study ART, or for antiretroviral therapy chest. And this is our conceptual model of how this might be beneficial. The chest system use, as I just mentioned, is designed to support intrinsic motivation. And this is really the evidence-based pathway for reducing relapse. And relapse is a well-known risk factor for reduced antiretroviral adherence. The piece that we're trying to study and build with this research is this other pathway, is that by using the chest system, which can capture social and behavioral data, we might be able to detect people who are at risk through the use of the ASK closer to real time. And this builds on some of the prior work that our group has done with shorter term EMA or ecologic momentary assessment trials with people who use drugs. And the theory here is that we can capture this data in real time and make this timely and support timely access to appropriate services, addressing that information chasm that I mentioned in the first slide. So the study is in two phases, and it spanned the two research groups that I acknowledged at the beginning at HIV, at Johns Hopkins HIV Care Program, where many of the patients in the NIDA-funded ALIVE cohort received care. We had an initial pilot phase with the target enrollment of 60 individuals that was intended to be a more intensive data collection period where we got people the app and we monitored them very closely with the goal of building a database that can be used to understand the predictive value of some of the variables. And the second phase, which is now underway and soon to wrap up, is more an implementation study in 4 HIV clinical practices here in Wisconsin, which is where I'm currently on faculty. The phase 1 study design recruited patients with HIV who participated in ALIVE, had a history of opioid use disorder, and a recent history of treatment failure. So this meant a viral load greater than 1,000 in the past year. So we were selecting people that we knew had sort of demonstrated them or had a track record of poor engagement in care, and were likely, although we would try to prevent it, to experience lapses in care during the study. The study design involved evaluations every 3, 6, 9, and 12 months, again, using the infrastructure of the community-based study of ALIVE. We provided a smartphone and provided training for the HS app for participants. The EMA component was actually limited to weekly surveys, not multiple daily prompts as we did in some previous studies, with an eye toward more sustainability of the engagement with the data collection. And there wasn't formal integration with HIV clinic staff. Again, we wanted to get this off the ground quickly to start building data systems to understand the prediction. And this slide shows some of our data that we collected. The heat map on the right shows what I think is a really interesting feature or deliverable from this product, which is we have this database where with a number of patients who churned rapidly between periods of being virally suppressed and non-suppressed. So green means good. So each row in this grid is a patient. If their box is green, that means during that month that they were assessed, they had an undetectable or suppressed viral load. And if they switch to yellow or orange, that's low-level viremia, and red is treatment failure greater than 500. You can see there's a large number, nearly half of people flipped at least one or more times between green and red. And those are the areas where we're really analyzing the CHEST, the EMA data to see what is happening and are we able to collect these behavioral risk factors in more real time. So, and we're starting to analyze those data, and there's a lot of different dimensions to the data, but I showed some of the basic findings here. The weekly survey that people use in CHEST was designed to use to capture some elements that are built around addiction relapse, but then also some things that are just hypothesized red flags or things that we, based on clinical experience and prior literature, know are associated with lapses in care. And it's injected drugs in the past week, did not have a place to sleep, skipped a meal because they didn't have money, criminal justice involvement, and then increases in anxiety and depression symptoms. And just for example, we showed here that people who reported 2 or more missed doses in the past week had a fourfold greater odds in having virologic failure to be in that red box. people who injected drugs were twice as likely to have a missed visit or a no-show visit. So these aren't big headlines. Like, we know that these are important risk factors. But what's exciting about what we've found here is that this relatively high-risk group that is poorly engaged in care and not with smartphone users, over the course of the year remained engaged enough to report these data reliably, and they have some predictive value. So that was an exciting thing to sort of see how it works, and sets us up, I think, for some next steps or some next studies. Another aspect that we will use the data that we're collecting through CHESS is in what ways are individuals who are using the system engaging with the app. I didn't go into detail of what the app does, but there are sort of different elements related to network communication. We code that as exposure or expression. Are people posting things on a discussion board? Are they just reading what other people have shared? There's a motivation sections where people do engage in journaling or expressions of gratitude, or that have been developed through previous studies. And we showed in our smaller study the degree to which people engaged in these, and these are just click, this is 27,000, it's a very high number. That's essentially anytime someone touches the screen in one of these sections, it gets logged. But this table just shows how these things diminished over time. So there was a fair amount of decrease in how highly people were engaged, but it remained remained substantial throughout the whole year. And some of them didn't decrease as many as others. For example, the weekly survey did not decrease very much. People were still, engaging in the self-monitoring aspects of the weekly survey. Some of the games and relaxations and the help with cravings features also did not decrease as many as some of the messaging or the discussion boards. So there's more to analyze there to understand how does this population interact with this kind of technology. And the second phase of the study is more of a pragmatic clinical trial where our goal is to evaluate the feasibility and preliminary effectiveness of the CHESS intervention on HIV care outcomes using a pre-post study design. The difference here is that it's over a longer period of 2 or more years. We are not providing phones. We're not incentivizing the use of phones. It's more to give people access to the service and see the ways in which they use it to support their own HIV care. The goals of the project were to figure out ways to formally integrate this. And one of the things that we proposed in the application was to grant providers access to CHESS data through a data dashboard. And this is something we actually didn't do. And it was a deliberate decision based on things we learned, which I'll get to in a second. But it learned some interesting things about how this technology works as when we try to bring it further to scale. These are the preliminary findings from the study. That's the phase 2 study, which is still underway. We had a fair amount of enthusiasm. Most people who were approached and screened were interested. We enrolled 208 people. We've had a modest amount of attrition, but fairly good as far as this population goes. Retention and the study-related aspects. The outcomes are frequency of missed visits and viral suppression. These are obtained from the EMR. And the intervention that are being used is moderated HS use and access to the weekly survey. So the framework, again, these are preliminary data. There's more to analyze here, but this is kind of the crude findings. We're planning to look at these outcomes of viral suppression and missed visits as a pre-post. So the top are the first 151 participants. We look at their viral load outcomes in the year before they enrolled in the study, for the 6 first months of the ramp-up period, and then afterwards. And we're seeing, without any test of statistical significance, a modest increase in the percent of viral load measurements that are undetectable or suppressed, from 80% up to 88%. We have a contemporaneous non-treatment cohort of people who were also identified by providers in the participant clinics as having a need for substance use treatment. And that shows there was also a modest increase over time. So this is the type of framework that we'll use to understand the visit, but we don't have final results to share. And another really important part of this study is thinking about, how do we use this type of technology at a systems level to improve aggregate outcomes. We've had anecdotal evidence that this works really well in the way that we intended. In the second phase of the study, case managers and providers, if patients were interested, could get notified of these red flag symptoms. Someone said they missed 3 doses. A study coordinator could send a secure message to the case manager, who would then reach out to the patient and get them engaged. So it works as in instances, and there's been some very rewarding success stories. But if we want to build this into a way that really supports practice on a larger scale, we need to have a pretty careful implementation research, and that's something that we're starting to scratch the surface of. We've collected sort of data from providers about how they use information and communication systems. We've had focus groups with care coordinators to understand how the sources of social support are given and how they work currently. And the participants in particular were interested in how the system meets their needs, how do they feel about the design elements, and with an eye toward further refinement and improvement. And this is probably the biggest lesson that we learned talking to providers. Everyone was very enthusiastic about our goal here. I showed that gap, the information chasm. Everyone says, yes, that information about drug use, about social determinants of health is critical. But when we asked them, well, would you like to get reports of these surveys in real time? They said no, frankly, is that they have information overload. They don't know what to do with it in real time. It varied by provider, but it was clear that if we want to harness this information, we have to do a fair amount of work to figure out how this information flows to support the delivery of care in a sort of coordinated way. And I think that sets us up for the next steps. So we're really enthusiastic and really grateful for the support of this project to show how the system can meet the needs of patients in our care, and are excited to take further studies into systems level adoption. We have now a R1 grant that's underway which is submitted, trying to translate these preliminary findings to this award to a hybrid implementation effectiveness study in 4 U.S. states. And we'll, over the next 6 to 12 months, be digging into this really nuanced and very interesting data from the population that's been collected by the app and some of the surveys. So I will stop there and be happy to take any questions. And thank you again for this fun opportunity to share our work.
Thank you, Ryan.
So, Ryan, I'm going to take prerogative, and since I can ask my question, and then we'll go to the ones that are in the chat. So this is great. And I know you've also looked at some provider level and maybe even community level factors that impact interruptions in care and quality of care. And I know some of your work has particularly focused on the level of stigma that providers have around people who use drugs and people who inject drugs. And I don't know if you have carried on any of that work through your Avenir Award, and if you have any more recent data or findings that you could just quickly talk about.
Yeah, I would say not systematic data collection, but the interviews that we did with providers that I alluded to. Stigma might be a role, but I think it's more of a sense of helplessness. Essentially the problems that we're trying to address through this work, a lot of physicians, providers see as social problems that I'm frankly not trained to address. So this needs to be someone else's problem. They're sympathetic with the need to it, but they don't feel like they're empowered or they have time to address them. So I think the way we did this, that I think the silver lining is here, is that we asked similar questions to a large number of people on a care team. And we really got support for that role, which I think is going to be key in future research, is the medical case manager or the patient navigators. They are hungry for this type of data and are very enthusiastic about hearing it. But again, trying to support team-based care is really the work. So I think we definitely found sort of a lack of enthusiasm among some providers. I don't know that it's directly caused by stigma targeted against the population we're trying to serve, as much as just not sort of maybe not my problem kind of sentiment.
We have a question that came through chat, and this is from Dr. Friedman, and we're trying to unmute so he can ask you.
Yeah, I think I'm unmuted. Yeah.
Okay, great.
Yeah, wonderful talk. Really suggests good ideas. When you presented some of your earlier slides, they showed that things like incarceration or becoming homeless or things like that might be risks to engagement. It wasn't fully captured in your theoretical model, but particularly with what you were just presenting about provider overload, would it make sense to develop contacts with community groups and an easy way to tie the HS data and what you're finding oriented towards evidence that maybe those things are going to happen, and then get community groups to provide support or training or even legal help to help prevent some of those?
Yeah, thanks for that question. It's a really important element. What's unique about our HIV care setting that we're working, it's not unique among all HIV care settings, but it's the community partner that I mentioned at the beginning, Vivint Health, has really adopted a patient-centered medical home type approach. So even some of the things you mentioned, like legal support, like they have those types of services in-house, or referrals to community-based organizations to help with some of these other things. The conduit to a lot of those things is that role of that care coordinator, that medical case management. Medical case manager who sort of sees as the person who coordinates access to these services. So really, I think you really nailed it. The idea that when those things are perhaps come to the awareness of the team, there is a fairly well-developed infrastructure for making those connections to community-based sources of support. I guess the problem that this project is trying to solve is how do you facilitate those existing connections in a better way? And we're lucky in HIV. People that have similar risk factors and have injection drug use problems who don't have HIV have a dramatically different landscape of services available to them. But in this HIV care setting, there's a lot that's already been built to meet some of those needs, and they're often unmet because of the sort of lack of awareness. So that's kind of what we're trying to target.
Yeah, in the Ukraine, we've experimented with having essentially community-based health managers. And if they were connected to something like the HS system, they could be proactive.
Yeah, that's a great suggestion.
All right. Still looking for any questions in the chat. But I did not miss anything from Q&A. All right. Fine, I don't see anything coming in, but I guess we'll just move on to the next talk in that case. Ryan, thank you so much. Wonderful talk.
Thank you.
And Redonna, you have something you want to ask?
Well, Kavi, it looks like we have a break now. We have a short break. Is that correct? Am I reading the agenda correctly? So can Aaron or Jeremy let us know, do you want people just to stay on but mute their phones for the break so that they don't have to sign back in? Is that your preference?
Yes. If folks want to just stay on, we'll put on some break slides, and we will return here at 11 o'clock.
And at 11, Dr. Sunil Solomon will give his presentation, and he is from Johns Hopkins University. So we'll just move right into his talk so we have more time for the science and for the discussion. Thank you all so much for attending. We have had as many as 85 participants, so I think we can consider this to be a great success. So we'll see you back and start at 11 promptly. Enjoy your break. Okay, are we ready to get started? Okay, you ready? Dr. Solomon, take it away.
Thank you, Redonna. So good morning, everyone. I want to thank the NIDA leadership for inviting us here today to share some of our data related to the Avenir grant. So my presentation is going to be a little bit different from the others because I want to give you a little bit of how I got the idea for the Avenir grant, what we have done in the Avenir, as well as what we're currently doing based on things we learned from the Avenir itself. These are my disclosures. So just to set the stage, because I know we have been talking a lot about HIV and PIVID in the US, but to set the stage and to bring everybody to the same page about HIV in India. So India is home to about 2.1 million people living with HIV. It is a largely heterosexual driven epidemic, but we do see concentrated epidemics among key populations. And as you can see from the latest Sentinel surveillance data, PIVID with a prevalence of 6.3% is the key population with the highest prevalence. For those of you who are also aware about India's unique position, we are located right between the Golden Triangle and the Golden Crescent, which are the two largest heroin producing regions in the world. And we have drug trafficking routes going through India from both sides, from the east and the west. And as you might also expect, India with 6 million opioid users is home to the largest number of opioid users globally, which should not be surprising because with a population of 1.3 billion people, we're home to the largest number of a lot of things globally. The most recent estimate for people who inject drugs in India is about 850,000 PIVID who live across the country. So I want to start by acknowledging the Indo-US Joint Working Group, which was really spearheaded by Jack Weizskaver and Jacques Normand from NIDA. So everything that I'm presenting here today is a result of 2 R01 supplements of $100,000 that it really has transformed our work in India. And the one thing we do know in India is how to stretch a little money and make it go a long way. So using these 2 $100,000 supplements, we were able to establish a longitudinal cohort of people who inject drugs in Chennai. We recruited 1,100 people, characterized HIV Hep C incidents, one of the first longitudinal cohorts in India. And simultaneously, we also demonstrated via a randomized clinical trial that more non-monetary voucher incentives do improve linkage to care and ART retention among people who inject drugs in India. So these 2 grants really spearheaded our work in India, but most of this work was focused in Chennai. And we very soon realized that PIVID across India faced similar challenges with access to HIV services. So we followed that up with another NIDA-funded cluster randomized trial. And the objective of this trial really was to look at the impact of integrated HIV service delivery, integrating HIV prevention and treatment services into integrated venues, which were PIVID friendly. And as you can see from this map, we were literally all over the map. All the orange spots indicate sites where we work with people who inject drugs. So I'm not going to go into this trial or its findings, but this is really what the baseline assessment of this trial is really what gave me the idea for my Avenir application. So as part of the baseline assessment, we recruited 1,000 PIVID per city across 15 cities in India. So we recruited a total of 15,000 PIVID over a year. So just to give you an idea of how respondent-driven sampling works. So this is the city of Kanpur in central India, where we really had no information on PIVID before we started our work there. So RDS generally starts off by identifying 2 individuals who are considered seeds who are highly influential. We give them 2 coupons, have them give it at random to any other 2 PIVID whom they may know in the city, have them come into the study. So when they come into the study, the recruiter gets an incentive and the recruit also gets an incentive. So it's a dual incentive mechanism. And when that first person comes in, we repeat the process, give them 2 coupons. And in this process, the PIVID population recruit each other in a city. So what I wanted you to look at this figure and really highlight is one is in most cities across India, we only used 2 seeds to recruit our entire sample of 1,000 people. The second thing I want to highlight is just the extremely high burden of HIV and Hep C in this population. Almost 65% of the population were infected with hepatitis C virus. But at the same time, I think what was most striking to us as researchers who were conducting a baseline study was only 3% of the 350 HIV infected PIVID in this population were actually aware that they were HIV infected. And none of the 650 PIVID who were hepatitis C virus infected actually knew they were infected with hepatitis C. So this really prompted the question of, can we leverage networks like the RDS networks to improve HIV case identification among people who inject drugs? So in my Avenir, we tried to adapt respondent-driven sampling from what it's traditionally used for. Traditionally, it's used for surveillance to try and assess disease burden or drug use patterns or something else. And we said, can we adapt it from being purely a surveillance tool to actually start delivering services? So this is not the first group to do it, but I think there are other groups that have also been doing it, especially the Aristotle group in Greece. So we were looking at 3 different strategies. The first one was time-based RDS. So the way RDS traditionally works is you set a sample size of 500 or 1,000, or you have process measures, and when you meet that, you stop. So what we said is, why don't we just let it run for a fixed period of time? And we decided this period was going to be 1 year. We implemented this in the city of Varanasi in Uttar Pradesh, which has one of the fastest growing PVID epidemics in India. It was estimated there are approximately 600 PVID who live in Varanasi. So over the 1 year period that we ran in Varanasi, we recruited 1,300 PVID, which was more than double the number of estimated PVID. Everyone was provided with HIV testing and all the positives were linked to care. So it really was a very efficient strategy of finding PVID and finding people who are unknown and linking them to the actual HIV program itself. The second approach we took was precision RDS. So the data from this contributed to the PhD dissertation for Alison McFall, who continues to work with us. So the hypothesis with precision RDS really was, there are recruiter characteristics that can predict the ability to find undiagnosed cases. As in, there are some people who by default were going to be much better at finding people who are undiagnosed compared to other people. So the approach we took was we built a prediction model using random forests and logistic regression. And then we randomized participants to either the standard coupon where everybody got the same number of coupons, or the precision RDS system in which people were randomized if they were good, in which people who were considered good recruiters by the algorithm got more coupons and people who were considered bad recruiters got fewer coupons. We implemented this in the city of Morinda in Punjab, which is again one of those really high prevalence PVID populations across India. So we used 2 characteristics to look at whether this was an efficient strategy. The first one was a number needed to recruit. So this is the number of people you needed to recruit to find one undiagnosed infection. So the lower the bar, the closer the bar is to 0, the better. And as you can see, the precision RDS did better throughout. It wasn't statistically significant. The difference was about 2.5 or 3 coupons fewer. But I think one thing that you need to understand is from an implementation standpoint, 3 coupons fewer translates to about $10 less. And when looking at a program which is trying to reach 1,000 drug users or 10,000 drug users, that translates to $10,000 or $100,000. So it really does impact program efficiency and program yield. The second indicator we used was the identification rate, which was a number of undiagnosed HIV-infected PVID diagnosed per week. So in this, what we want to be is we want to be the higher bar. And so as you can see, the precision RDS approach did significantly better over the standard approach in terms of the identification rate. So based on these 2, it is safe to say that the precision RDS does appear to be more efficient than standard RDS approaches as a case identification strategy. But one thing we always recognize through RDS is what 50% of the coupons that go out never come back. And so we always wondered where these coupons went to, whether particular pockets or particular parts of the city where the heavy injectors were living, where you couldn't really bring them back into the venue. Could we actually supplement this RDS with field-based testing by going to these different places? Our hypothesis really was prevalence of HIV and prevalence of Hep C is not going to be evenly distributed across the city. There are going to be hotspots with a high burden and some hotspots with a much lower burden, and targeting high burden hotspots will be a more cost-efficient strategy. So our focus really has been from an implementation standpoint. We implemented this strategy in the city of Gorakhpur, which is also in Uttar Pradesh at the foothills of the Himalayas. We recruited 500 PVID and we plotted out the prevalence on the map. And as you can see, just as we expected, there were about 4 hotspots where the prevalence of HIV was significantly higher than all the other hotspots. So we were about to start processes when COVID hit, And so we've stopped, and now we're currently redesigning the RDS Plus to make it RDS Plus Plus, which also integrates COVID restrictions, COVID precautions. We've just got permission from the IRB to restart our activities. So this data is definitely coming in the next couple of months, I hope. I think the one thing about the pandemic is none of us know exactly where the pandemic is going. So I think the Avenir really did provide us with a lot of answers about service delivery, but it raised a lot more questions to us about transmission itself. For example, we were reaching all these people, getting them tested, but why was incidence so high? Like, I haven't presented this data, but in some of these cities, the incidence was high as 10 or 12 per 100 person years. And as I said, like, RDS doesn't really recruit transmission networks. It's built on the principle that you recruit randomly from your networks. So it's not necessarily your transmission networks. And it also doesn't really connect people across different networks. And while I showed you that the prevalence is different by different spaces, it also doesn't incorporate the role of spaces in the transmission. We felt that if you really wanted to prevent onward transmission of HIV and Hep C, we really need to understand underlying network structure, its overlap, its space and time, which really prompted us to start another NIDA funded grant, which is called the Spatial Network. So this was a longitudinal cohort of PVID in the city of New Delhi. The objective of this trial really was to understand the role of egocentric, sociometric, and socio-spatial networks on HIV and hepatitis C transmission. I'll explain what we mean by these different networks for those of you who are not very familiar with network recruitment strategies. So the way we built this cohort was we started off with 10 index participants. The index participants were asked to name the people whom they injected with in the prior 1 month. And they were also asked to give us like descriptive characteristics, unique identifying features of these people, and only those specific individuals were allowed to come into the study. So it really was that specific injection network within the prior 1 month. And when those network participants came in, they served as the index for the next wave of recruits. And we kept doing it until the entire network was saturated, then we added a new index. So the one thing I want to point out here, which is different from RDS, was in RDS, we would say, give it to any 2 people whom you wanted, versus in this study in Delhi, we actually had them give us specifics about the name, the description, and a unique identifier or appearance of how this person actually looked like when they would come in. The other challenge of working in India, as you may imagine, was the unique identifier or the description for most people was he short and with a mustache. So it was getting really hard for us in India to try and distinguish one short person with a mustache with another short person with a mustache. And so we actually incorporated something called a fun fact, where we allowed them to tell us something about this person, such as he is originally from Bihar, his name, his wife's name is Jyoti, he has 2 children, or something along those lines. The other thing we also did was by design, we allowed duplicate participants to come in for the trial, but we didn't, just to establish the cross-network linkages. So to give you a sense of what the recruitment looked like, when an index participant came in, we asked them to name the people whom they immediately injected with. So that would form the egocentric network. Then we asked those participants, did they inject with anybody else? And if they didn't inject with someone else, that would also be the sociometric network for that particular index. And this is another example where the egocentric network is essentially the same, but when we ask them if they injected with someone else, it's like a much larger network. And so the sociometric network ends up being a little bit different. And then we also asked people to give us specific locations where they injected. And we considered everybody injecting within a particular location to be one spatial network. And similarly, there were other spatial networks in Delhi. And over time, we asked them, did you inject with the same people? Did you inject with different people? Did you inject in the same location? Did you inject in different locations? And as you can see, over time, people move, people in migrate, people out migrate, and the network structures also change accordingly. So this is essentially how we characterize the different networks. So and so to give you a quick snapshot, so those 10 indexes recruited 2,512 people who inject drugs in New Delhi. About 75% of the coupons came back in, but what is really striking was the HIV prevalence at baseline was 37%, and only 8% of them were virologically suppressed. The hepatitis C antibody prevalence was 65%, and only 20% of them had cleared the virus. In terms of HIV incidence, though the study really was looking at transmission, I think what was striking was the extremely high incidence, especially given the baseline prevalence. We were only talking about 63% of the cohort that was not infected at baseline. And in that cohort, we had 159 new infections during longitudinal follow-up. And with a similar thing, we also saw with hepatitis C. So the HIV incidence was 22.3 per 100 person years, and the hepatitis C, this was antibody seroconversion. If you include HCV RNA, clearance and reinfection probably is going to be a lot higher. So because the focus really was sociospatial networks, so this is what the sociospatial network looked like. So this is part of the dissertation of Stephen Klipman, another doctoral student from Hopkins. So as I mentioned, we did start with 10 indexes, but 5 of these indexes merged into one large network in the middle, which seemed really hard for us to understand. So we actually went out to the site, and what we found was there was this one particular location in New Delhi where on any given day, at least 1,000 people cycled through that location. There were a lot of migrant workers, a lot of migrant laborers who all lived here. So I know this is a very beautiful figure and it's very complicated, but I want to take you through 2 specific situations of what exactly how complicated this is. So if you look at this one red incident infection, this is like the classic incident infection you would expect, where he's connected directly to another incident infection, 3 prevalent infections, and 1 person who's HIV negative. But if you look at the second case, so this person has 4 connections in his egocentric network, none of whom were detectable, were HIV infected at their prior visit, but he was only 2 steps away from several people who were HIV infected. So in terms of predictors of HIV incidence, we looked at the standard individual level predictors, as you would expect, age, recent injection, and injection frequency were strongly associated with. But what we really were interested in were network level predictors. So the first thing we looked at was your egocentric network. So this is the people who are in your immediate circle. So for every person in your immediate circle who was HIV infected with detectable virus, your risk of HIV seroconversion went up by 31%. And as I showed you in that second example, there was no one in your immediate circle who was HIV infected, but you could still be connected through someone who was HIV infected, either 1 step or 2 steps or 3 steps away, which is what we tried to assess with your sociometric or the path distance. So what we found there was for every additional step between you and someone who was HIV infected with detectable virus, your individual risk of HIV seroconversion dropped by 40%. So these definitely do show you some signal that HIV infection, that HIV viremia does play a role in transmission, even among people who inject drugs. But I think what was really striking was like that injection. So we ran a machine learning model, and we found that this one particular location was associated with a 3.4 times higher likelihood of seroconversion. And this association held good even after adjusting for egocentric and sociometric factors. So what I want to show you in this slide is what this network actually looks like and how it evolves over time. These purple nodes reflect the hotspots, and these different nodes of these different colors reflect different participants. So what I really want you to focus on are these red nodes and these red edges or these lines. The edges connect nodes with each other and nodes with hotspots. And what you can really see in this figure is over time, this network just evolves, and that big purple spot in the middle, that's pretty much where spatial network hotspot number 40 is. And as you would expect, you see a lot of clustering of infections close to that spatial hotspot number 40. And what we found was like 113, like 70% of all these incident infections reported injecting at one particular hotspot. And when you're looking at socio-spatial distance from that particular hotspot, so if you were in, even if you were not injecting in that hotspot, if you were injecting with someone who was injecting in that hotspot, there was a definite risk. So the further you were from that hotspot, your risk of HIV seroconversion kept going down. So our next steps, we have completed whole genome sequencing. So we are trying to look at this to look at, does U is equal to U also hold good in people who inject drugs? And the other thing we also want to do is we want to recreate these networks using purely phylogenetic data without any of the self-reported data. We have been able to resume our visits post-COVID. So because we have this rich network data, we want to look at the transmission of SARS-CoV-2. And for those of you who are following, there was a lot of migration and lockdowns in India, which sort of serves as a natural experiment in India itself on terms of network structure. But so far, the spatial network study is only in one city in India. Now, how does this fit into the larger elimination agenda? So with the last two slides, I just want to show you something that we were able to do with funding from Abbott, where we sequenced the core 5 UTR region from Amritsar, Delhi, Kanpur, and Imphal to look at clustering of infections. So here you can see there's a lot of intercity clustering. For example, the cluster in Delhi had infections from Kanpur, Delhi, and Amritsar, whereas the cluster in Amritsar had infections from Kanpur, Amritsar, and in Imphal. We were also able to conduct a Bayesian phylogeographic approach and reconstruct the geospatial dynamics of HIV. The temporal information was obtained using time-resolved phylogenies. So as you would expect, the oldest mean common ancestor in India was in Imphal. It seems to have moved from Imphal to Kanpur all the way to Amritsar. So the size of these circular polygons really are your Markov reward counts. So essentially showing you that most of the local epidemics are in Imphal and Amritsar, and Delhi and Kanpur serve more as transitory points, which is consistent because Imphal is a much older epidemic closer to the Golden Triangle, and Amritsar is a much newer epidemic closer to the Golden Crescent. So what does this all mean? So from the Avenir and the earlier work that I've showed you, networks can definitely be leveraged to improve access to services, but we are just beginning to scratch the surface of networks, because the networks are a lot more complicated than just interpersonal connections. And to really interrupt transmission to achieve HIV/AIDS epidemic control or the HCV elimination, we really need to take a step back and look at these epidemics from a much larger lens. I'd like to thank a lot of people, and I think especially NIDA, for all their funding support over the years. And I think one thing I do want to highlight is, while we are doing research, a lot of our research really is service delivery and it's implementation research. So over the years, we've provided services to over 50,000 people who inject drugs in India purely through NIDA research funding. I want to end by acknowledging and thanking one of my site teams, which we have many of in India. Thank you.
Thank you very much, Sunil. Excellent talk. We know we have to address gaps in HIV care cascade to be able to, address the HIV epidemic and the HIV epidemic. And so the more ways we can reach out to these people who are falling through the cracks, not able to reach the services, it's better for us to know this and to address. And thank you for all this important work you're doing in India. And now let's look at the questions. There's Dr. Friedman. He asked, there are a lot of similarities between your and Dr. Friedman's research and interventions, so we should communicate on it, he says. I guess that is your collaboration for future. In particular, his Ukraine colleagues have done good work with artificial intelligence to improve case detection for HIV. So I think it's just an opportunity for collaboration. There is a question from Jason Blakard. Is HIV incidence associated with having HCV? Is HCV incidence associated with having HIV? Are baseline untreated viral loads higher for HIV when HCV is also present?
Thanks, Jason, for those questions. So we have not looked at the viral loads untreated in HIV mono-infected versus co-infected, but we can definitely take a look at that. In terms of the association between HCV and HIV, so HIV incidence does not predict HCV incidence. As I mentioned, for us, we do see most of the HIV seroconversions, if they were HCV antibody negative at baseline, they seroconverted for both. I don't know if that's the question you're asking, if it's mono-infection seroconversion versus dual infection. So if that is the question, then yes, people who are antibody negative for Hep C, and when we see them as an incident HIV infection, 90 to 95% of them have seroconverted for both hepatitis C and for HIV. It's very rare that we see HIV seroconversion without hepatitis C seroconversion. I'm not sure if that answered the question.
I think that's part of the question, Sunil. I think he also is asking, do you notice any differences in viral loads when there is a dual infection with HCV?
Right. So I think that I've answered. Like, we haven't really looked at it, but I'm happy to definitely look at it, and I can get back to him on that.
Great.
And I also want to thank Dr. Friedman for his, I think I did see one of his team's presentations in Tajikistan last year where they used artificial intelligence. So I think there really is definitely a role for collaboration.
So Dr. Volkow is asking, are there distinct position of women, there you go, of women versus men in the networks? Do they have less centrality?
So there definitely is a role. So the epidemic in India, generally in most of India, is completely driven by men. For example, in this cohort, we only had about 25 women, and most of them are in the periphery. Whereas if you go to the Northeast, we haven't done this work here, but I would expect like distinct networks of female people who inject drugs that would exist by themselves in the northeastern states of India.
Great, thank you. There is Steve Shropta. He has a question about opioid overdoses. Steve, are you unmuted? You can ask if you want. So he's asking, do you have thoughts as to whether opioid overdoses map onto HIV network incidence cases in India?
Yeah, and related to this question, because I am now unmuted, you know, these in US, we have syndemics. So we have these intertwining epidemics between opioids, HCV, and other things. Can you talk a little bit, as well as overdoses, about things like mental health disorders, STDs, maybe the need for primary care within this group as you think about intervention?
Sure. Thanks, Steve. So we have collected the information on opioid overdose, but we haven't looked at it specifically in connection with incidence and prevalence. But I agree with you. I think the way we've been designing most of our programs in India really has been around healthcare needs of people who inject drugs. I think it's very hard to run a program just for HIV or just for hepatitis C, because most times you really have to figure out what is that hook, what do they need? And the goal of the program really is improving health. So we do have these models right now in India, which again, thanks to NIDA funding, we are running these integrated centers which do primary healthcare, harm reduction, HIV, and we've just started a trial where we're integrating hepatitis C services. So I think really the model is building a one-stop shop, which is not disease specific, but person specific, and really building in social support, building in like economic opportunities, homelessness support, food, nutrition. I think those really are the drivers for PBID communities globally, not just in India, but I think that's like a uniform theme globally. And we generally tend to look at PBID with a disease lens. We need to step back and look at it from a person lens and be like, if I was a PBID, what would I want? And really build our programs around that. I think that's where we are sort of lacking. And I think once we switch our focus and realize that once we put on this people lens, it really will make a much bigger difference.
So we're running out of time, Kavi, and I see there's some other questions. Maybe people with specific questions or comments can reach out to Dr. Solomon directly. Because I want us to be able to go to the next talk since it's 11:30. So Michael Newcomb is scheduled to present now, and he is from Northwestern University. So take it away.
All right. All right, well, thank you very much for inviting me to give this presentation today on my Avenir Award, which I received in,
Hang on just one second.
Yes.
Can you go over to display settings and flip it because we see
I'm sorry. Yes.
That's okay.
Actually, I've got a solution. Please hold.
Yeah, just flip it around so we get your slides big.
Oh, technology these days, it's killing me. Better?
Better, perfect.
All right, thank you. I apologize. As I was saying, thank you for inviting me to give this presentation today on my Avenir Award, which I received in 2016. So I'm currently in my final year of this award. And the title of my talk today is Using Relationship Education as a Platform to Reduce HIV Risk in Young Male Couples. I first just want to thank and acknowledge my team, including my co-investigators, my incredibly talented staff, NIDA for the receipt of this award, as well as some other funding that I've received to support this program of research, including an R01 from NIAAA and the Sexualities Project at Northwestern University. So just by way of brief background, why focus on couples in HIV prevention amongst young men who have sex with men in the United States? In the late 2000s, a bunch of data emerged indicating that a large proportion of new HIV infections in the United States were occurring in the context of primary partnerships or serious romantic relationships, and that this was especially pronounced amongst young men who have sex with men. One estimate that up to 79% of new HIV infections were occurring in the context of primary partnerships. Intuitively, this makes sense as you start to think about it. In serious relationships or primary partnerships, people tend to use condoms less frequently. There's a higher likelihood of engaging in receptive anal sex, which carries higher risk for HIV transmission. And quite simply, you have sex with the same person over and over again. So if that person is a person living with HIV, there's a higher risk of HIV transmission if viral load is not controlled. More recent data has suggested that it's not just condom use that tends to decline upon relationship entry, but one of the primary predictors of PrEP discontinuation is entry into a relationship. On top of that, a large proportion of HIV positive YMSM under the age of 25 are not aware that they're living with HIV. So this creates a bit of a perfect storm in terms of HIV transmission, that if young people who are unaware they're living with HIV enter into relationships, they then reduce their use of protective behaviors and make it highly likely that they would transmit HIV to their partner. So interestingly, right about the same time that I started in on this line of research, this is a photo of my now husband and me, I met my now husband. So relationships and relationship skills were something that were very much on my mind at the time as I was thinking about how to address HIV risk in young male couples. And in digging into the relationship literature, one of the most robust effects in that literature is what's called the marriage effect, which is that upon entry into a serious committed relationship or a marriage, multiple aspects of health and well-being start to improve. That includes mental health, substance use, various other aspects of cardiovascular health, on and on and on. However, one of the factors that can impede the health promotive effects of being in a relationship is stress. So if individuals in relationships or couples are experiencing stress, then that reduces the positive influence of being in a relationship. And for same-sex couples, there are a whole host of other stressors that we must face, both as individuals and as couples, that compound the effects, the negative effects of stress on our day-to-day lives. So that just means that it's all the more important that we help young people to build relationship skills in order to optimize the health promotive benefits of being in a relationship. So that led, or the combination of my work as well as my personal life led to the premise that optimizing relationship functioning is a strong platform through which we can promote both relationship health and other aspects of individual health, including reducing HIV transmission risk. And that led to the current project, integrating relationship education with primary and secondary HIV prevention for young male couples, or the Together intervention. In brief, Together is a 4-session hybrid intervention. It's a hybrid group and individual level intervention for both HIV negative and HIV positive coupled YMSM. The intervention consists of first 2 group sessions that are attended by anywhere from 2 to 8 couples and live facilitated by 2 facilitators that focus primarily on didactic skills with regard to relationship skills and sexual health promotion. And then those 2 group sessions are followed by 2 individualized skills coaching sessions in which each couple works with 1 facilitator to apply the didactic skills to the unique experiences in their relationships. Broadly speaking, the skills that we promote in Together, first, the relationship functioning skills. We heavily emphasize building effective communication skills, as well as coping skills that couples can use as a dyad in order to navigate stressors that they encounter, either as individuals in their individual lives or together as couples. And then the sexual health intervention, which actually comes at the very end of this intervention. Primarily, this intervention actually focuses on relationship functioning, but the sexual health component is 3-pronged. So first, we focus on building and maintaining sexual satisfaction and pleasure within the dyad. Next, we focus on building and maintaining relationship sexual agreements, which are another word for monogamy or non-monogamy arrangements, which vary according to the couples and the preferences of the couples in our intervention. And then finally, layering on top of the unique needs of each couple's sexual agreement, primary and secondary HIV prevention strategies. So with regard to the current project, we first conducted a very small non-randomized pilot trial, which served as the preliminary data for the submission of this DP2. And based on that, I was awarded this grant to evaluate Together in a comparative effectiveness trial of substance-using young male couples, 200 couples in total, or 400 individuals, with 12-month follow-up. Couples are randomized to either the active condition, the Together intervention, or the control condition, which is based on existing public health practice for HIV prevention and care for couples. So that would mean either couples-based HIV testing and counseling for those who are HIV negative, or the Life Steps medication adherence and risk reduction protocol for folks who are living with HIV. And then serodiscordant couples would receive both of these interventions at the same time. In terms of eligibility criteria, so you have a little bit of a sense of who these couples are. In order to enroll in the trial, couples need to be both assigned male at birth and currently identify as men, both aged 18 years or older, and one partner needs to be aged 18 to 29. We allow partner two's age to be 30 or older in recognition of the fact that having an older sexual partner or romantic partner is a unique risk factor for HIV transmission amongst YMSM. Both individuals need to identify one another as primary partners. They had to have had anal sex with one another within the 3 months prior to baseline. And at least one of the partners needs to report condomless anal sex with a serodiscordant or unknown status partner within the last 3 months. And that partner can be either their primary partner, if they're serodiscordant with their primary partner, or an outside sexual partner. With regard to substance use criteria, at least one of the partners has to have reported a binge drinking or drug use episode within the last 30 days. So right off the bat, we experienced our first challenge with this grant, which took the project in a very different direction than what I anticipated. It was actually a good problem, which was that I received another grant just a few months after I received this DP2 award that was also planning to recruit 200 young male dyads in Chicago with nearly identical inclusion criteria. So I immediately freaked out in a good way, but freaked out nonetheless, and realized that it was not going to be feasible to recruit 400 couples from Chicago in this exact same time period. So it started getting me thinking, Could this intervention be done entirely online via telehealth? So not, changing it into an automated e-health intervention like an app or something like that, but could we just simply do the same intervention with couples across the United States over a platform like Zoom? So I was able to work with my program officer, Rich Jenkins, to come up with a plan to do this. First, we conducted a brief pilot study with 15 dyads in January of 2017, which was very successful and only resulted in us having to make a couple minor changes to the implementation of the intervention. So first, the group sessions, which can last up to 2 hours long, can be very difficult for somebody to sit through online as opposed to in person. So we split those 2 group sessions into 3 group sessions, which means that we now have a 5-session intervention. And in order to reduce the length of those group sessions even further, we pre-recorded a lot of the didactic content that's reviewed in those sessions, sent it to our participants a week ahead of time for pre-review, so that then the group sessions could focus entirely on discussion and activities amongst the couples. In terms of the enrollment process that we use for this study, it's quite complicated in doing this with couples. It starts off by having each member of the dyad complete an eligibility screener. Once the couple has deemed to be preliminarily eligible, we complete a couple conversation, a couple confirmation call with each member of the dyad separately, which essentially is asking specific questions about their relationship and partnership in order to identify fake couples. After that, couples complete the baseline process, which includes a baseline survey, a self-video recorded communication task, and then remote completion of STI testing for urethral and rectal chlamydia and gonorrhea. After that, couples are randomized to either the active or control condition. The second challenge we faced fairly quickly was recruitment and enrollment of couples. As you can imagine, we learned a hard lesson very quickly, which is that recruiting couples is really difficult. But if done correctly, it actually has very high payoff, which I'll demonstrate in some of the subsequent slides. And we have indeed completed recruitment, randomized all 200 dyads. However, one thing that we observed is that there's fairly large loss to follow-up between initial eligibility screening and actual randomization, and larger loss to follow-up than we see in other types of behavioral trials. This is for a number of reasons. First, it can be difficult to engage both partners in a study. So often one partner would be interested and the other person would not. It's very difficult scheduling multiple couples into group sessions. And so sometimes in trying to work with people's schedules, couples would be lost to follow up because we just couldn't place them into a cohort. Also, we found that there were a number of folks who were in partnerships with transgender and non-binary folks, which according to our eligibility criteria were not eligible for the study. We also found changing eligibility over time, given the fact that there is an extended period between initial eligibility screening and actually taking the baseline survey. More specifically, HIV risk behavior would sometimes change from eligibility screening to the actual baseline survey, which would then make them ineligible for the study. We also observed a fair amount of couples dissolving their relationships between eligibility screening and randomization. So for some of those couples who were entering into this in some amount of distress. And we currently have a grant pending with NIAD that I'll talk about at the very end that aims to address all 5 of these issues. In terms of study progress to date, as I mentioned, we have enrolled and randomized all 200 dyads or 400 participants. The enrollment period was March 2017 through December 2020, with a bit of a pause on enrollment during the initial stages of the COVID-19 pandemic. All participants were recruited online, predominantly through social media advertisements, as well as referrals from other colleagues who were recruiting participants online for different types of studies. And here's where the payoff, I think, is really most highly illustrated, is that although it's very difficult to engage, to get couples to the randomization point, once you do, there's incredibly high retention at follow-up. So although we're continuing to follow up with our couples across the 12-month follow-up period, we currently have a 96% retention of our participants at 6 months and a 91% retention at 12 months. Also, in terms of other indicators of progress, we've published 4 manuscripts with 3 more under review, all using baseline data from this fairly large sample of dyads. In terms of demographics in the sample, there is a slight majority in the sample of our participants being a non-Hispanic white. We also have fairly large representation of Latinx participants and somewhat lower representation than we had hoped for Black and African American participants, which is the third challenge that I'll note here. This was for a couple of reasons. First, during the final months of our anticipated enrollment, the COVID pandemic hit. And this was when we were really hoping to focus all of our enrollment on people of color. The other thing that's important to note is that our other study of couples that's focused just in Chicago has a much higher representation of Black participants. I think one of the main reasons for this is because with us being based in Chicago, we have a lot closer ties to community-based organizations in the Chicagoland area that serve to legitimize our research enterprise in Chicago. And so we hope in the future to be able to build stronger partnerships with national organizations working with people of color in order to improve enrollment. Other aspects of demographics important to note is that just about 20% of our dyads had at least one partner who was known HIV positive, and it was a majority gay identified sample. Also interesting to note here that more than 10% of our sample was never tested for HIV in their lifetime, which is interesting given that our mean age, even when you take out partners who are age 30 and above, is about 25 years of age. We also have really good distribution across the United States, except for the Plains states. So this shows where our couples are coming from, and strong representation across the index of relative rurality, which includes both rural and suburban MSM, which are highly underrepresented in research on men who have sex with men. The largest proportion of our sample came from the South, followed by the West. You might anticipate we would have a much larger proportion from Illinois, but as I mentioned before, we also have a study ongoing with couples in Chicago. So all Chicago-based couples, of which there are currently 127, are shunted into that study. I'd be remiss if I didn't talk briefly about substance use at baseline. So we have a fair amount of heavy substance use, about a quarter of our sample in the moderate to high range for risk of alcohol use disorder, the same for risk of cannabis use disorder, and about a quarter reporting use of some other illicit drug in the last 30 days. The primary substances of use here are cocaine, ecstasy, and methamphetamine. Substance use is also a very common theme that came up in sessions as impacting both communication within partnerships and sexual health. And the research questions, once we gather all of our data around substance use, are what is the impact of this intervention on alcohol and drug use in general, as well as how does substance use impact the efficacy of the intervention on HIV risk reduction. In the interest of time, I'm going to skip over the impact of COVID on this project, but as you can imagine, it did delay us a bit. And I want to focus on our interim trial analyses. So we conducted interim analyses of the active condition only about 9 months ago in order to support a grant submission. The findings are very promising. So we see at 12 months, there's a significant reduction in STI infection, that's chlamydia and gonorrhea infection, a significant reduction in the number of condomless anal sex partners, a significant increase in PrEP use, as well as amongst PrEP users, a 100% PrEP persistence at 12-month follow-up. And we also see that certain aspects of relationship health also significantly improve. Here, specifically, the use of positive communication skills. And the next steps for Together, I mentioned that we currently have a grant pending with NIAID. We submitted a grant for the D-LIGHT initiative, or the Digital Limited Interaction Trials and Epidemiology Initiative, that are large-scale digital clinical trials testing the effects of digitally delivered HIV prevention interventions on HIV incidence. We proposed a hybrid type 1 implementation effectiveness trial of Together, received an impact score of 30, and we're cautiously optimistic about funding of this award in 2021. And as I mentioned, this proposal was specifically informed by the DP2, specifically designed to address the various barriers that I noted throughout this presentation. So in this project, we aim to eliminate group sessions and harden the didactic content into an automated e-health program in order to get more couples in the door and reduce loss to follow-up. To enroll both dyads and individuals, so allowing individuals who are in relationships to enroll, as well as single people to enroll in anticipation of future relationships. Enrolled dyads, people who do come in as a couple, will still receive skills coaching sessions in order to evaluate their additive benefit over and above the automated program. We're adapting the interventions to the needs of transgender MSM and non-binary folks assigned male at birth who partner with cisgender men. And we're more comprehensively engaging with organizations across the US to better reach MSM of color and Black MSM in particular. Thank you very much for your time, and I will end there.
Thank you, Michael. Great talk, and congratulations on your R01 in the queue, I guess.
Hopefully, yeah. Yeah, it's pending U01. So yes, I'm cautiously optimistic, but can't say anything for certain.
Right, we keep our fingers crossed. So let's see if we have any questions come through here in chat.
So while you wait for questions, I have one comment. So if you become a part of that NIAID cooperative agreement, make sure that you continue to have your collaborative partners look at substance use and remind them of the important role that substance use plays in achieving virological suppression as well as in prevention.
Yeah. Yeah, it's absolutely been a central theme that we've encountered throughout this intervention. And, I didn't mention we're also doing HIV testing as part of the intervention. So during the intervention, they receive HIV testing and also a 12-month follow-up. We actually, our last couple, both of them tested positive for the first time in the study. And just as a reminder of the fact that these data on transmission and relationships are not outdated. In I think almost every case in which somebody tested positive, substance use or alcohol use was a major theme that those folks were struggling with. So very relevant to NIDA.
And also, as we remind NIAID, very relevant to NIAID.
Yes, that's a yes.
I'm sure Nora's laughing at me by now.
We also co-fund several initiatives with NIAID. This is for general audience and also Michael. So we are partnering on the LIGHT initiative.
Yeah.
So,
yeah, and I'm excited. The scale of the D-LIGHT projects are larger than most behavioral interventions are, and that's designed purposely to be able to detect effects on HIV incidence. But it also has a lot of implications for substance use that sometimes we're limited in our behavioral trials and the ability to detect effects on the use of certain substances because of power issues. And in this case, with such a large sample, there's a lot of opportunity to look at moderation effects of specific substances of use on intervention effects, to look at the ability to reduce the use of those substances, both in individuals and dyads. So it's exciting to be able to collect data at that level.
Well, if you have any questions for Dr. Newcomb, please reach out to him. So I think we will just now move on to the final talk in the series. Thank you again, Dr. Newcomb. Redonna.
And the final presenter is Dr. Zachary Klase from Drexel University. So take it away.
Hello and thank you. Sorry, let me just move some stuff out of the way. So what I wanted to start with today really is a thank you to the grant, sort of an overview of some of the things we've gotten from this. You know, publications as a measure of output, I think is pretty important. I mean, 5 publications could have came directly from this project. I'm going to tell you about, you know, pieces of about 3 of them here. And then of course, a number of collaborative efforts that, you know, might not be directly related to the grant, but you know, the capabilities we built for the grant lent themselves to helping others. And of course, the stability of long-term funding, it makes it a little easier to say, yes, I'll help you with that project. The most important thing for me, you know, in my mindset though, is training. And the grant here got us to train 2 postdoctoral fellows who went off and got the careers they wanted to do. We minted 3 PhDs, an MS student. I have 2 wonderful graduate students still in my lab. And because of my previous position where I started this grant was a primarily undergraduate institution, we got to bring 12 undergraduates into the lab and show them a lot of molecular biology of HIV. And then also, you know, grew a number of collaborations with people around Philadelphia and around the country. Most maybe pressing to me, you know, got a promotion out of this from assistant professor to associate professor because of the work done here. And then something I'll give you a little bit more info about later is we actually moved the research lab from University of the Sciences to the Drexel University College of Medicine. I'll tell you more about that later. For now, I'll just say that if you've never moved a research group in the middle of a worldwide pandemic, you are missing an experience. So what was great about this mechanism is you come in and you don't have aims. You just present to the study section, you know, here are the big problems that I think I see in the world, and here's how I think I can deal with them. Excuse me. So this is actually a figure that I showed to the study section when I sought the grant in the first place. And it's talking about the idea that, you know, we have HIV as a disease and all the things that come with that. We have substance use, and we understand that a large number of HIV positive individuals abuse substances, so that worsens the disease. But that in my view, we also need to think about attempts at therapy and HIV cure as another overlap in the terms of problems that we're having here. And the red places were things that I was particularly interested in, and the keys were places that my lab was already making inroads into these problems. What I'm going to talk to you about today, because obviously this is a very big problem still, is the issue of benzodiazepines as a substance of abuse and how they might fit into HIV cure. And that actually relates to the mechanisms of latency and persistence of HIV. So when we started this project, what we knew was from a paper that I published just as I founded my lab, you know, almost 7 years ago now, in which we had identified this RUNX1 protein, which is a important T cell specific transcription factor that actually inhibits transcription of HIV, and therefore we thought might have a role in latency and persistence of the virus. And we were able to identify, you know, both that the HIV promoter, as the blue bars here, are responsive to when you put more RUNX into the system, you see a suppression of transcription. And we were able to map one particular site, site 2 up here on sort of the end of the LTR as a real RUNX binding site, because when we mutated it, the effect goes away. That in itself is just nitty gritty in molecular. What was most interesting is we got a drug out of it. Our collaborator, Dr. Liu, was interested in RUNX inhibitors because they're useful possibly for treating AML leukemia. And he had identified this compound out of a Roche library called RO53335. And RO5 on its own wasn't too exciting in terms of activating the HIV promoter, but we saw when we mixed it with SAHA or varinostat, which is another sort of leading, compound in the quest to reactivate and clear HIV infection, we got this synergistic activation, and that was very exciting. So the Avenir then really started with work from my first PhD student, Wei-Am, and she looked at this structure of RO5 and she says, well, that's a benzodiazepine. This is a class of compound that might be some of the most well-studied compounds in the chemical library. Benzodiazepines have been in the clinic for 60, 70 years now. They're pretty well studied. Why don't we go and look at some of these clinically used benzodiazepines, things like Xanax, which is alprazolam, and Valium, which is diazepam, and see what they do in the system. And so she went back to the original assay and whether or not we could activate the HIV promoter and kick cells out of this latent state using all of these different benzodiazepines. The orange bars here are the synergies with SAHA, SAHA alone being here. So maybe the surprising thing was that every single one of the benzos that we tested appeared to have some functionality in increasing SAHA activity, which in itself was exciting. Like, okay, maybe we can improve this other drug. But then when you look at alprazolam and diazepam here, the blue line is the drug alone. So they're having this great effect on the HIV transcription without SAHA. So they're doing it, all by themselves, which is really kind of exciting, as a possible intervention in therapy because these things are already in the clinic. And this works across multiple dose ranges, even down into the, sort of lower end of the clinically used range of the drug, which was very exciting to us. So at this point, I should, catch us up, right? Because I think this talk is sort of completely different than what we've been hearing so far, and remind everyone that HIV is really dependent and has a real, sort of importance in the chromatin state, in the epigenetics of the cell. And what I mean by that is, we've got DNA in our cell, and that DNA is packaged in an organized fashion into nucleosomes. And that's DNA wrapped around a set of histone proteins. And that's the basic structure that makes up, the chromosome and helps regulate, function in the nucleus. And when you look at a nucleosome, and I have one over here on the right, access to this DNA and transcriptional activity of that DNA is controlled by how tight this interaction is between the histone proteins and the DNA, and by what you recruit to these histone tails sticking out the side. And that's what's really, the basis of controlling all gene expression in the cell. And because HIV integrates itself into our genome, it's at the mercy of this process as well. So we wanted to know, in the latent state, this virus that we're trying to clear out, what is it that alprazolam and these benzos are doing to the epigenetics, and how does that figure into transcriptional control? And then the next star of our presentation becomes Angel, one of my current PhD students. I think she's actually got a benzodiazepine on her shirt here. And she used chromatin immunoprecipitation assays to look at these epigenetic interactions. And she started with this model of, here we have DNA wrapped around nucleosomes, and in a transcriptionally repressed state, like when HIV is off, there should be transcriptional repressors and there should be methylation of the core histones. And then Runx figures into this because we think it sits up at the end of the HIV promoter and helps drive all of this. The prediction being that when we inhibit Runx function, that we should switch to an active state. We should see acetylation of histones and recruitment of transcriptional activators. So Angel started by looking at acetylation of histones. And maybe not surprisingly, the three benzos we looked at, our original RO5 compound, alprazolam and clonazepam, they don't do anything to acetylation. And that's really, probably not too strange. That's not what they're supposed to be doing. The histone deacetylase inhibitor SAHA, that does increase acetylation. But it gets interesting, and then alprazolam, our winning drug so far, increases that acetylation to a greater degree. So why is that? Well, the literature told us that there's a connection between the RUNX1 protein and the STAT proteins, especially STAT5, which has been shown to be critical for HIV transcription. So we wanted to look at STAT5 with the idea that when RUNX isn't there, that STAT5 recruitment would be increased. And sure enough, now we start to understand what it is that alprazolam does and why it's different than these other two benzos that we tested. Alprazolam alone can help recruit STAT5 to the HIV promoter, presumably through kicking RUNX out and allowing STAT5 to come in. And then that's even better in the presence of the histone deacetylase inhibitor. STAT5 then, and you can tell this is sort of a complex layer of Russian dolls, as it will. then recruits CBP-P300. This is a histone acetyltransferase, driving some of these acetylation events down here. And we see that alprazolam also increases that. So we think, alprazolam is inhibiting RUNX, which allows recruitment of STAT5, which brings in CBP-P300, and that explains the increase in acetylation that we see up here. Now, Angel then asked a really good question. She says, okay, we've been looking at HIV, the promoter specifically, but we've just shown that this, prescribed medication also has an effect on STAT5. Is that just specific to the HIV promoter, or is this something that's more broadly happening in cells that are exposed to alprazolam? So she took cells that don't have the benzodiazepine receptor. And I think that's key, because that means the effect we're looking at here is something other than, the classic effect of the drugs. And she treated them with our 3 benzos. And she did a Western blot looking at STAT5 levels, and they don't change. But then she looked at the levels of phosphorylated STAT5, which is the activated form of the protein. And she sees that alprazolam, but not the 2 other benzos we tried, increases phosphorylation. She then did a follow-up experiment where we could track phosphorylation of STAT5 using a phosphotag, which retards movement of the protein through the gel. So as you get movement of this protein further up the gel into these sort of smears on the right, that's increasing phosphorylation. And we see a nice dose-dependent effect of alprazolam on phosphorylation of STAT5. So it does seem that alprazolam is having a broad effect outside of what we see with the HIV promoter, sort of suggesting that it could be doing other things in the body, even outside of HIV patients. So maybe this is a good point to remind everyone what that is, right? So JAK-STAT signaling, right? Signaling through Janus kinases and phosphorylation of STATs is the signaling step in response to nearly all of the cytokines and interleukins. And in case of many of the really important ones, STAT5 is the major signaling protein after you get receptor engagement, which means that STAT5 is helping to control the immune response, right? Because we're signaling to the immune cells and to the body using cytokines that signals through STAT5. STAT5 engagement and activation then leads STAT5 to go to the nucleus and turn on a lot of these things as well. There's sort of a feed-forward loop on things like IL-2. And that is because STAT5 is pictured down here at the bottom. STAT5 is an adapter that's actually helping to recruit a number of epigenetic modifying proteins. I talked about CBP-P300 as it relates to HIV and driving acetylation of things, but it also recruits histone deacetylases and methyltransferases, meaning that STAT5 itself is something that's altering the epigenetic landscape. And if we're turning on STAT5, it's going to affect potentially the expression of a lot of genes, and sort of near and dear to my heart, the whole inflammatory process as well. So to look at this directly, we found a cell line reporter system that's used for measuring IL-2 expression. And it does that because it has an IL-2 receptor on the surface, which activates the JAK kinases, which activates STAT5 and turns on a reporter. So we wanted to use this to see if we could just sort of skip the IL-2 step and directly activate STAT5 and really prove that alprazolam is having an effect on STAT5 directly. And so we did that. Our positive control is IL-2, but you can see a nice dose-dependent response to alprazolam, really driving home the point that we're acting on STAT5. Now, at this point, you might start to go, wait, you're telling me that alprazolam could be a stand-in for IL-2. Do we need to worry about this therapeutically? And I'll point out that these low bars down here are what are in the sort of therapeutic range of the drug. So no, I don't think we're going to get, replacement of IL-2 effect in a human being taking alprazolam, but we wanted to check that, of course, right? Because T cell activation is something that's both a bad outcome for a patient and important for knowing the activation state of things like HIV. And I won't walk you through this whole figure, but we did pretty extensive testing looking at T cell activation in response to all the agents we've been using to mess with HIV transcription. And the answer is that, as you would expect from something that's been used for a long time and is pretty safe, alprazolam isn't activating T cells. But the idea that it could be helping STAT5 activity really led to the question of whether or not this might be potentiating immune response in a proper setting. Like, could alprazolam be immunoprotective? Could it be something that would, when the T cell was going to respond anyway, could it actually help and give a better outcome? And so we wanted to look at that. And one of the ways we have been looking at that is by looking at intracellular cytokine expression in CD8 cells. So this is the cytotoxic T lymphocyte response, which is critical to clearing viral infection. And we think would be one of the things that we would need to actually, clear an HIV infection if we were going to do this reactivation strategy. So we took white cells, immune cells from HIV patients who had been suppressed on therapy for at least 6 months. We exposed them to antigen, so GAG peptides, the major structural protein of HIV, and treated them with drugs to block release of cytokines so we could measure their intracellular levels. And this is just showing you an example flow plot of how we gate this out and what we expect to see in terms of interferon response within CD8 T cells. Now it's still early days. We're working on increasing our N up to another 5 or 6 patients on top of this. But I'd say that the results from our first 3 in our pilot are pretty good. We compared alprazolam to the histone deacetylase inhibitor SAHA, to vorinostat. And the reason there is, this is a drug that people have been wanting to use to reactivate HIV, but it hasn't been successful yet. And going back probably a decade, work from Bruce Walker's group showed that that's because histone deacetylase inhibitors actually suppress the cytokine response in immune cells, and that would be bad as a curative strategy. And the statistics aren't there yet, but the trend certainly is going in the right direction, supporting this idea that SAHA is decreasing the level of interferon expression in response to HIV antigen. What's great is alprazolam is then actually higher than that. Looks like it might actually be sort of trending towards higher than the control as well, which would support that idea that maybe this would be a way to improve immune response when you need that to happen. The great thing, if we could ever get there in terms of statistical power, would be to show that you could use these two together, and that alprazolam might reverse some of the bad effects of SAHA. We also looked at IL-2 expression, because IL-2 dysregulation is another part of the problem with histone deacetylase inhibitors. And as has been reported by others, we see this increase in IL-2 expression when we use SAHA, and we don't see that with alprazolam. So again, I think things are coming up roses, and I'm hoping this will be, interesting as we recruit more patients and find more samples to work with. Now, sort of, in the last couple of minutes remaining here, I want to give you some insight into why I think this is important in a bigger sense. And, benzodiazepines they're a therapy, right? We prescribe them to people. We prescribe them to something like 75 million people in the US every year, which is a fairly large number of people. Depending on the survey and data you look at, about 10% of the US population has abused a benzodiazepine, and maybe 1% actually qualify for dependence, right? So this is an abused substance. And all the problems that go along with abused substances are seen with the benzos as well, including increased risk of HIV seroconversion. And I believe something like 30% of opioid overdoses actually have benzos involved in them as well. So it is a problematic chemical and an abuse issue. And the data I just showed you would suggest that benzodiazepines, in addition to their effect on the CNS, are actually driving epigenetic change, which, especially if you think about it in the terms of the CNS, means they're driving pretty permanent changes in the cells that are exposed to them. And we wanted to come up with a way to look at that, both for benzos and other abused substances. So a former postdoc of mine while he was with me, Luca, who's now a senior scientist in the HIV discovery program at Merck, came up with this assay wherein we take the nucleus out of a whole cell, we strip away the nuclear membrane, and that leaves us with something that in my mind looks like those wicker balls you see in hotel lobbies, in that we have this cage that contains the nuclear components within it, but now has large openings in it. And what that allows us to do is we can then just use fluorochrome labeled antibodies to do live staining. So we can look at nuclear architecture without the need for fixation and actually follow changes over time. And this has allowed us to take what I think are some actually pretty stunning sets of images showing nuclear architecture in the cell, and allow us to sort of compare that to what has been shown and known before in the literature, and identify where areas of active transcription are, and look at actual changes in structure and not just amounts or location like you might see with CHIP or Western blot. It's interesting also because we can use this to do live imaging, right? I told you that this nucleus isn't fixed. So we can actually watch changes over time in response to things we do to this nucleus. And as an example of that, here is a nucleus that we have stained for histone acetylation. So all these bright spots you see are presumably areas of active transcription. And I'm going to show you 4 hours of time compressed down to 30, 40 seconds. And 1 hour in, we're going to add the histone deacetylase inhibitor SAHA, and you can actually watch what this does to acetylation in this nucleus. So here we are. We add the SAHA to it. You can see that it gets brighter as the level of acetylation goes up. And then there's actually a reorganization of the acetylation. So it actually moves to the periphery of the nucleus. And this fits with ideas on transcription, with the idea of where you would see the most active transcription. And maybe the easiest way to think about that is if you transcribe something, you want to pass it out of the nucleus. So you do it sort of close to the edge. But we can see these changes happening over time. We've started to apply this to the issues of substance abuse. And we've taken, what I'm showing you here, are astrocytes that we've exposed either to SAHA or to morphine as our stand-in opiate, or the combination of the two. And maybe the real interesting part of this is if you just look down at the bottom, the green staining is showing you histone acetylation as a marker of gene activation. When you treat with the histone deacetylase inhibitor, the level of acetylation goes up. That's what SAHA does. Morphine, you don't get a huge change, but you do see some sort of active areas of transcription at the edge of the nucleus, suggesting that yes, morphine is turning on gene expression. Most interesting to this and relevant to HIV, cure strategies is it seems like morphine-treated cells are resistant to the SAHA effect, which really, gives us something to think about if we're saying that we're going to use histone deacetylase inhibitors as part of a cure strategy. They might not work in the high percentage of people that are currently using opioids. We've been following up on this opioid issue in both trying to model over time, look at chronic exposure to morphine, and also look across the doses. And what we've seen is that it's actually the smallest amount of morphine that drives the largest amount of change. So I've got 5 nanomolar over here on the right, as opposed to half micromolar here. That's where we see the greatest increase in acetylation. And we see this really unique architectural change as well, where the acetylation goes from being sort of diffused throughout the nucleus to being lined up at the edge, and also very perinucleolar, which for those of you in the crowd that love epigenetics in the way I do, you're going to realize that that's strange. Perinucleolar chromatin is thought to be the stuff that should be off. That should be heterochromatin that's kept in the middle. So not only do we see aberrant acetylation of histones, we see an aberrant placement of those acetylated histones, really suggesting that something strange is going on here. So, putting this all together, I mean, for HIV cure, I think we should be looking at RUNCs and as a way to improve HDAC inhibitors. For the substance abuse side and the sort of interesting in what we're doing to patients as we prescribe things, we need to realize that alprazolam is probably a RUNCs inhibitor and is modulating things like STAT5 and possibly immune response as a result of that. Some things that I didn't show you is that this also actually moves the placement of the nucleosomes on the DNA, which is great. And then the big takeaway being that I think we all appreciate that drugs of abuse are a confounding factor in HIV as a disease, but they're also going to be a confounding factor in cure, and we should be paying attention. And the last thing, just to thank the program, is what's great about this is the flexibility of not having to chase down the aims you promised to do. So here's what we're going to do in our final year. This is just a scheme showing that we can isolate useful CNS cells from flash frozen CNS tissue. So we have a request in with the NNTC to get human brain tissue from HIV patients, either with or without opiate abuse. And we're going to use the microscopy to come up with a big atlas of epigenetic architecture. We still have a pie in the sky thing ongoing where we're trying to come up with a way to visualize specific genes, i.e. the integrated HIV. I think our winner right now is to use a CRISPR-Cas-based system, and we're kind of putting this together in a piecewise fashion. And that would allow us to map where HIV is on top of our nuclear imaging. And then the newest collaboration coming out of some work with Peter Gaskill is to sort of move this transcriptional control work, the most of which is done in T cells, into myeloid cells that really are important for CNS infection. And that collaboration with Dr. Gaskill is one of the things that came out of my opportunity of moving the lab to the Drexel University College of Medicine. because the Pharm Phys department at Drexel really has this, I'd say, small but mighty core of NIDA-funded researchers and a really large amount of expertise coming out of our chair, Dr. Mayuchi and PJ, who I just mentioned, and not to downplay their expertise and their strengths, but I want to highlight Dr. Jackie Barker down here as a recent DP2 recipient who hopefully we'll be hearing from in this format in years to come. Also want to, of course, thank my lab, the large number of people we've trained from this, and you for listening to me, and I'd be happy to answer whatever questions may have come up.
Thank you very much, Dr. Klase. That was really interesting talk. I think it'll be cool to do some in situ work that you are planning to do to get some contextual data in terms of what types of cell types, that are, do you see effects that are in a cell type specific manner? Because the data you showed is with astrocytes. So I would be really curious to see what happens with microglia and monocytes.
Yes, so would I. I mean, that's been we've been sort of slowly building up to make sure that we're going to do this right, sort of because of the preciousness of the samples that we're asking for. So overcoming the hurdle of getting the correct cell types out was a big one. And we've been working with some of the computational people that I have upstairs at Drexel, and we now have a way that we think, like one of the limitations with the microscopy, of course, is you're looking at one cell at a time. We've taken test sets of images and fed them to neural nets, and the computer can identify the same differences that we thought were there and confirm them across like 800 nuclei or something. So the idea is going to be to scan in, huge amounts of data, and then in a hands-off way, allow the computer to tell us what the differences in shape are. And I think that's really the only way we're going to be able to accomplish this, because we need to have a high N to account for human variability, all the different cell types, the various conditions we want to get, and then to really get to the data. And I'm very excited, and I'm glad we have a year left to get it done.
Yeah, I mean, please do look at other NIDA opportunities. It's interesting work. We want to keep you going. Congratulations on that. And then let me look at what questions we have coming in. There's John, can we unmute John? Maybe you can ask the question. So he's asking, what do you think is going on with the perinuclear staining? Because it is.
I don't know. I'm trying not to get too excited about it, because that's, acute exposure in tissue culture. And I think the images there are U87s. So they're sort of a much maligned astrocyte model. We haven't done that yet in like the primary astrocytes we've been working with. So I'm waiting to see if it holds up in real samples before I get too excited. That being said, I don't know. What I can glean from the mapping we've done so far is that, there's these established regions of the nucleus where you expect to see certain modifications, and we see that. I think naively, based on, how we do chip assays, you sort of expect, okay, when I see acetylation, I expect to see the acetyltransferase with it, right? Because that's what put it there. In the architecture, that's not necessarily true. It seems like the enzymes that are responsible for the modifications are actually held in the other compartment. So you have an inactive compartment. The acetyltransferases are there so they can acetylate the histones, and then they get shuttled out to the other active compartment. So perhaps what's happening there is a deficit in the transit of the genes and the rearrangement after you get the change in the modification. But that is an extreme guess.
I also have a question from Dr. Volkow. So she's asking if, okay, I hope I can pronounce this right, if aldosterone effect not mediated by the benzodiazepine receptor, have you identified the target?
Right. Yes, and then I see a follow-up question about the naloxone sensitivity. We haven't done naloxone yet. What we have done is all the epigenetic work has just been done in cells that shouldn't be responsive to benzos in the classical way. So I'm fairly certain that the effect we're seeing has nothing to do with the classical anti-anxiety effects and signaling through receptors. That implies then that you're getting entry into the cell, and that's been established for RO5, I think, fairly well. We have started to do some mapping with our wonderful chemistry department downstairs, and I didn't show them. I didn't have the time, and they're a little bit beyond my ability to explain quickly. But we've done computational mapping, and there is a binding pocket on the RUNX1 protein that accepts the benzodiazepines. One of the things that's probably setting alprazolam and diazepam apart from all the other ones as a class is there's a second binding site on the other face of the molecule that appears to accept only them. And I think we'll find out, we have to actually map out the residues, but I think we'll find out that that's the face of RUNX that's important for the STAT5 interaction, and the drug is acting sort of directly on that complex.
So we have one more question from Dr. Valentino. Are morphine effects naloxone sensitive?
Are like the morphine effects on acetylation? That we have not done yet. I think you're going to find that there's some hint that SAHA and morphine both have an effect on the same drug transporter. I know some of the early work on SAHA, they had to reformulate it to get it to cross the CNS because it was, shooting itself in the foot in terms of drug pumps that were getting it in. And I think that's probably what's happening. We haven't done the test to see if naloxone would change it.
I think Rita also has a subsequent question. Can we unmute? Maybe Rita can ask. There. Go ahead, Rita.
I'm unmuted. Okay, I think you answered that question. It was if alprazolam isn't producing its effects on STAT through the benzodiazepine receptor, right? Because you did it in the cells that didn't have the receptor. What protein is it interacting with? And I think you addressed that question.
Yeah, I will say I think it's still a useful experiment. I mean, T cells likely have the peripheral receptors for benzos, right? And so, it could be an additive effect.
Right. Yep. Thank you. A great talk. Thanks.
So thank you all. Thank everyone for all of your wonderful presentations. The strength of your science is demonstrated in the fact that while Dr. Volkow had a very busy schedule, she stayed for the entire half a day and was participating in most of these discussions. So Nora, let you close out the meeting. I'm thrilled that you were able to be here, and it just speaks to the quality of your all's presentations and the quality and innovation of your science.
Yeah, no, Redonna, I was supposed to be in 2 meetings today, apart from this one. I didn't exaggerate when I said that they had triple scheduled me, but I just couldn't stop watching the science. And I think that's really the excitement where we want to generate research that's so unexpected and yet gives us clues of how things work with one another. So I really was mesmerized by the talks that I heard today. I want to congratulate all of the presenters. And I also was fascinated by the diversity of the topics and the creativity and innovation in each one of them. So thanks to also for you and Gabi for overseeing this really amazing program on the Avenir. And the Avenir, we chose the title Avenir because these are the stars of the future. And I would sort of say they are already the stars of the present. So it's wonderful to see.
Thank you, Nora.
And I was actually in the room many years ago when the lights went on in your eyes and we were talking about something and you said, let's do an Avenir to go with avant-garde. And spoke about the title and what it would mean. So it's a real privilege to be able to be here now at this juncture and work with Kavi on this program and see it come to fruition. We will be doing a similar half day for our avant-garde awardees that will be taking place in late April. We'll be sending out a lot of mass communications around that. So we invite you all, everyone who's participated today, to participate in that presentation as well. And again, if you have any questions about applying for future funding, please reach out to program or reach out to us. We want to maintain all of your really important high priority work. You're just doing amazing things. So thank you, everyone.
Thank you.
Bye-bye.